have a huge role in transmitting the research information. The journal editors should ensure that the ethical conduct, methodological quality, and data credibility of their accepted manuscripts. Post-graduation and continuing professional education should provide a balanced, unbiased information about new devices, because the postgraduate students are not sure when they use a particular product or a new technique whether this is they are not sure about the harm and the benefit. That should be addressed. The dental clinicians should develop a reasonable level of knowledge. That is very, very important on the methodological aspect of research, so that they can apply these research findings into their practice. And the patients should be comprehensively informed on the effects of new therapies and techniques. We should not hide information from patients. They have the right to know about all these things, and they should be provided the sufficient information regarding their health. And dental companies also cannot shy away from exercising their responsibilities. They have to provide strong levels of scientific evidence to support the introduction of new materials in the market. As clinicians or researchers, we should ask the dental manufacturing companies or the dental companies to provide sufficient information on the new products. So the conclusion, the planned translational research is decisive for impactful clinical advances in dentistry. Basic researchers, clinicians, scientists, and clinical scientists and clinicians should work as an integrated team to achieve their goals. And better integration of the available evidence in our clinical decision making is a need of the hour. So with this, I end my presentation. This is the institution from where I come from. I am working in this institution as additional professor, Government Dental College, Kottayam. This is my email ID. Those who want to interact with me can email their queries. I may not be able to answer most of your queries, but I'll try to find out the answers from experts and I'll try to respond to you. Anyway, thank you one and all for the patient listening. I think it's almost more than an hour since all of you have logged into this presentation. Thank you, Department of Periodontics, Pushpur Dental College, for inviting me to this session. I'm actually delighted to be part of this program. Thank you, one and all.
Dr. Baiju, it was a very informative lecture from your side, and really you have covered a lot of things, especially the highlights of the topic is oral diseases related as come to evidence-based, followed by you have covered even how it's connected to social media and other things, and you have regulatory bodies also you mentioned about, and definitely by which I agree with you that how the clinicians has to translate the work to the bedside. With
Thank you
this,
so much.
that topic is open. Congratulations, sir, and a very, very interesting topic you have taken, and most of the people definitely will do the research mode. They will take up higher research. That we can open the topic right now, Dr. Shireen and Dr. Nebu. For the discussion.
Yes, sir. Dr. Baiju, can you hear me?
Yes, sir. Very good evening, sir. Good morning, sir.
Good morning, sir. Good morning, Madam Chambert, sir. I am Dr. George Orgis.
Okay. Oh, namaste.
Namaste, sir. Namaste.
Okay.
Dr. Baiju, you have presented the topic very well. Congratulations. It was very clear and lucid. There is no questions I have to ask, no queries, but one comment I would like to make. It is regarding the procedure surgeons or our doctors doing procedures without adequate clinical evidence. As you have said, especially you touched upon the socket shield technique and the use of or removal of the buccal pad of fat for, and that's very good. But very often we are forced to do things, because sometimes the patient will ask, or some of our juniors or even the postgraduates will ask, sir, this is such a technique is there, why don't you? If you don't do that, they will think that we are still preventive or we are not adequately informed. And there are situations when we have to do such technique which don't have practical or the other adequate supportive evidence. Just a comment only. Very good you have told about that point. Something to you would like to add to that, sir, Dr. Baiju?
Thank you, sir, for actually spending so much time in listening to my talk. First of all, I know you are a very experienced clinician and very experienced academician and administrator. Sir, the point that you made is a valid one, but sir, without at least a robust multi-center clinical trial and sufficient number of patients, actually we are not supposed to introduce something new to a patient, even if it is a very simple cosmetic procedure, a minor deviation, even if the patient consents to it. If everything goes well, it is fine and good, no issues, no questions asked. But some mishap happens, then nobody will be in our side because the regulatory bodies, the governmental agencies won't be backing us. So it's a safe practice as an academician or as a clinician to look for sufficient evidence, that to quality of evidence. At least you should have a randomized controlled trial with good number of patients to show that this works in patients. The harm is, sorry, the benefit is more than the harm. If that is established, you can go ahead. Otherwise, it will be risky even if the procedure is a minor deviation in the incision or a new suture technique. Even a simple things, it will be risky unless you have sufficient evidence to back up. Thank you, sir.
Any questions in chat box? Dr. Sharing or anybody can help?
Thank you, Dr. Baiju, for the comments. Thank you very much.
No, sir. No questions yet.
Dr. Baiju, I just strongly agree with you what you mentioned about the supportive data required to do any work. I strongly agree with you because
Thank you, sir.
system of data collection is we have to improve in India, so that we can definitely
Yes.
we can make more research and we can publish more articles, or
Yes.
we can come to the
Yes.
bedside to, I mean, as you said, the bedside we can do the usage of the materials.
Yes. Thank you.
We can conclude with Dr. Sherry.
Yes, sir. So thank you, Baiju sir and Sham sir for this session. Baiju sir for this enlightening session on the various problems associated with oral health and translational research, and Sham sir for moderating the session. Thank you very much. Now moving on to the next session, I invite
Thank you so much.
Professor Dr. Eben sir, Professor Dr. Eben Thomas, Professor and Head of Department of Oral and Maxillofacial Surgery, Pushpari College of Dental Sciences, for the moderating the next session.
Thank you, Dr. Soumya, and thank you, organizers, for having involved me in this interesting research-oriented conference, the two-day bioradiance, the first of its kind, where we have focused upon translational research in tissue engineering as the theme. I can see a very huge number of participants, in excess of 300. That's indeed a good sign, showing more and more people interested in this field of research. I can see attendance from dental college, medical college, engineering college, and even other institutions. The credit again goes to the person who actually spearheaded this bioradiance, our own dear Reverend Father Matthew Marinji Lachin, who's actually a visionary, started off with this Pushpay Research Center, PRC, about 10 years back, and never stopped looking back. And now is come to this particular state where we were able to induce interest in many people in the field of research. We've had two scintillating sessions on various aspects of this translational research. One from Dr. Remya from Sabita Dental College, who spoke about very well about the various aspects of research, translational research, and how this data can be transformed to the patients. We also had another one talk on about eminent speaker, Dr. Baiju from Government Dental College Trivandrum, who have give us an in-depth knowledge about the gray areas or the lacuna in the field of transit research. Now we have in line our own dear Dr. Nandakumar Kavli, another one world-renowned eminent scientist in the field of nanotechnology and nanosciences. He's a senior professor in the School of Pure and Applied Sciences, and he was a former director and joint director of International and Inter-University Center for Nanosciences in Mahatma Gandhi University. And I'm sure we're going to have a very good session from our own Naduma sir. Over to you, sir, for your presentation.
Thank you, Doctor. At the outset, I would like to thank PRC for inviting me to give a small presentation to share my views with regard to the role of government and educational institutions in the context of translational research for a better world. I shall try to share my slides now. So the title of my today's talk is Role of Government and Industry in the Support and Regulation of Translational Research. Is my slide getting changed?
Sir, could you put it in the presentation mode, sir?
I've put it in the presentation mode, but is it coming out in the full screen?
Not yet, sir. Not yet.
Uh, I really don't know, but sometimes I have this issue. So shall I proceed with the present mode of presentation with the slides on one side? Is it okay? Because I really don't know how to fix it. I'm not in my institute at the moment. I'm near to the airport to fly to Kolkata. So I'm in a hotel.
Uh,
Is
Sir,
it okay if I present like this? So this is my first slide. Uh,
yeah, yes, sir. Just try to click that, sir. Is it coming? The presentation.
is the second slide now coming?
No, sir. So could you please click on that slideshow?
I put it in the slideshow actually.
Okay.
In the slideshow,
Or in the toolbar on top, can you just click on the slideshow and on the toolbar on top, after
yeah,
animations, you'll find a
yeah,
slide.
yeah.
So just click
Let
on
me
that
see.
and then can you just start? Yeah, from beginning. Just try
Yeah.
that.
Oh, it's okay.
Not yet. So okay, sir. We'll continue with this.
Yeah, I'll continue like this because I'm yet to fix it because of the shortage of time. I will proceed like this. So when we talk about translational research, I would like to make a point that most of us are pursuing research in one way or other, but how far we were all successful in translating our research in the laboratory to a societal requirement level, that is at the application level. So most of the time, our research uh, is restricted to a level at which we are more interested in publishing all our data. Apart from that, we hardly go for patenting of our inventions and then translate our research findings. So, in general, if you look at translational research, which is basically meant to produce more meaningful, applicable results that directly benefit the human society. Since majority of the audience are related to health, I will be trying to talk more of the health aspects in this present talk. But at the same time, I will try to point out some of the important aspects related to other fields such as energy as well. So the major objective of any translational research is to translate basic science discoveries more quickly and efficiently into practice. So that is, we will do the benchmark research, and then from bench to bedside in the case of translational research in the field of the medical sciences. And then we need to translate that to the patient care level, that is at the clinical practice level, and then the translation will proceed further to population interventions, that is at the practice levels of the medical doctors. So this is very, very important. The translational research has become very important and thereby to encourage and promote multidisciplinary collaboration among laboratories and clinical researchers. It also incorporates the desires of the general public, with the communities being engaged to determine their needs for health innovation. That is, on a global perspective, we need to have certain kind of solutions for the benefit of the society in general. Particularly, I stress upon the fact that we need to concentrate more on the lower economic strata of the society. And therefore, in the translational research, we need to identify and support the adoption of best medical and health practices. So you need to have the research aspect along with the general practices which we need to implement among the patients. And at the same time, we need to have certain kind of policy matters to be considered to have a full-fledged translational research. So in general, any translational research involves a transition process, which in turn turns all the observations what we have obtained in the laboratory, clinic, and then thereby to have community interventions that will improve the health of an individual or a public in general. So, in general, I can say that the translational science is the field of investigations which basically focus on understanding the scientific and operational principles underlying each step of the translational process. So, when we talk about the translational research, the first and foremost thing we need to consider could be the challenges we may face in the process. The foremost aspect is the lack of coordination and connectivity between research and a clinical team or clinical teams in general. And then comes the funds. So we require quite large amount of funds or money to implement our ideas through a project, which will undergo certain critical stages in the research process. And most of the time, in general, I should say that we may lack novel ideas to design the clinical trials. And there is also the cross therapy area applicability of MOAs and targets, and thereby we need to have a sort of repurposing of our ideas. The culture of translation and shared goals of fundamental research for humanities will become another challenge in the process. Another important challenge we may all face is the lack of proper data availability, which we can correlate with the success rates and real time examples through the implementation process. And if more teams are involved in the process, there could be intellectual property right conflicts, there could be financial conflicts of interest, and at the end, we may also face certain ethical issues with respect to resource allocation basis, the nature of the research, chances of success, and impact at the end. So these are the basic challenges we may have in the translational research process. What are the drivers of translational research? The foremost aspect is to ensure the success of any research and development, increase the research and development productivity, and shorten the research and development life cycle. The drop in the research and development productivity has pushed the industry to improve the success rate of R&D programs. Improve R&D productivity by optimally utilizing existing knowledge, collaboration with other industry players, and taking novel approaches. So this is a major challenge in the entire process. For example, if you want to connect with an industry, they might have certain kind of objectives because industry in or industrial personnels are in general profit oriented. They need to have the product in a very short time period in the market. So that will become a very challenging aspect for any research and development partners. Need not be within the industry, but it could be with a research organization. Repurposing and lifecycle management is another drive we need to keep in the process of translational research. Increasing life expectancy and evolving disease pattern. This is in particular with regard to aging population and rapidly growing life expectancy in most world populations, which have resulted in an increased prevalence of the chronic diseases. For example, in the last 2 years, we were all facing the COVID-19 situation, and how far we were all successful in translating the entire research and development pertaining to COVID-19 prevention of chronic, COVID-19 disease in the global perspective. That is still a debatable kind of discussion, which I'm not going into that aspect, but I will try to uh, give more thrust on what kind of translational aspects we can think of in the process. Promise and expectation of growth in healthcare is always there, but at the same time, the continued rise in prevalence have resulted in the projected growth of healthcare spending. Despite the pricing pressure, treatments are getting costly due to the prolonged use of medicines and hospital stays. So these are some of the factors which may affect the translational research aspect. And coming to the different stages in translational research, which is normally uh, 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 termed as T spectrum, as I have mentioned, the T0 stage, which involves the basic research. In the case of healthcare sector, we have the preclinical and animal studies defining the mechanism, targets, and lead molecules. And then we will go to the T1 stage, that is the translation to humans, which involves proof of concept, phase 1 in clinical trials, new methods of diagnosis, treatment, and prevention. And in the T2 stage, the translation to patients, that is phase 2 and 3 clinical trials, which involves controlled studies leading to effective care. So these three aspects are basically the translation from basic science to human studies. And in the T spectrum, when we come to T3 phase, that is the translation to practice, which basically termed as phase 4, clinical trials and clinical outcome research, which will deliver the documented and timely care to the right patient. And in the T4 stage, we will try to translate this to a larger community. That is a population level outcome research, which will a true benefit to the society. So you have the translation of a new data into the clinic and health decision making, which is very, very important. And we can also look at the international scenario with respect to translational research initiative. For example, the Bill Melinda Gates Foundation, in association with the National Institutes of Health, has become very successful. And the Creative Spark, that is the Higher Education Enterprise Program of British Council, and of course, FDA, US Food and Drug Administration, is a major key player in the translational research. If you look at the translational research initiatives in India, we have the BIRAC from the Biotechnology Industry, that is the Biotechnology Industry Research Assist Council, a Government of India initiative. And the Department of Science and Technology is always a key player in this field. And we also have the ICMR, Indian Council of Medical Research, through the Stanford India Biodesign Scheme. And we, of course, we have the Ministry of Health and Family Welfare and Department of Biotechnology, Government of India. There are different government organizations which support translational research with international partners. I'm very happy to mention that we have been trying this BIRAC kind of proposals for quite some time, but very unsuccessful in getting such kind of programs through. So BIRAC, Biotechnology Industry Research Assistance Council, is basically to foster innovation and entrepreneurship. They intend to promote affordable innovation in key social sectors, empowerment of startups and small and medium enterprises, contribute through partners for capability enhancement and diffusion of innovation, enable commercialization of discovery, ensure global competitiveness of Indian enterprises. So this is a very interesting uh, uh, partner at the government level. The Stanford India by Design was launched in the year 2007 as a first of its kind collaboration between Stanford University, the All India Institute of Medical Sciences, AIMS in New Delhi, and Indian Institute of Technology, Delhi are partners in the initiative. So each year, 4 Indian fellows came to Stanford to spend 6 months working in multidisciplinary terms to learn and apply the biodesign process to real-world healthcare projects identified in Stanford's hospitals and clinics. The program is now known as the School of International Biodesign based at AIMS. And there are quite a large number of international research grants for translational research. A few among them are Duke RIT Collaborative Translational Research Grant, Medical Research Council Research Grant, NIDCD Research Grants, Stanford Culture Translational Research Grant Program. In India, we also have certain number of translational research grants, such as Accelerated Translational Grant for Commercialization, Contract Research Scheme by BIRAC, Attract Life Program, Sri Ramakrishna Paramahamsa Research Grant for Translational Biomedical Research. So Government of India is now putting lots of efforts for the translational research. If any of the international projects or national projects, if you submit, it should have a strong component of the possibility of translational research and commercialization. Unless until we involve, unless until we specify and uh, emphasize that particular part, no grant will be given to any researchers nowadays. If you look at the Accelerated Translational Grant for Commercialization, which generally encourages technological innovation, DBT basically envisages providing funding opportunities for fundamental research that is explicitly aimed toward application development. The major vision of such an approach is to support translational research in Indian academic institutions in the cutting edge areas of biotechnology for application development. And similarly, Ministry of Human Resource Development has also started a new initiative, such as the Scheme for the Promotion of Academic Research Collaborations with the rest of the world. So MG University is having currently 7 Spark projects, the only university in the state of Kerala to have 7 Spark projects in its first initiative itself. So that is an opportunity given to the researchers to interact and collaborate with researchers across the globe at the translational level. And the contract research scheme by BIRAC, this is a very nice scheme which applies innovation as a coherent plan to deliver academic capabilities of translational research toward product development. Any academic institution may be the primary applicant with one or more partners, of which at least one is a company with a DSIR recognized in-house R&D unit. In most of the calls nowadays require an industrial partnership with an R&D unit. With such a vision, MG University has also started a company within the campus. Apart from its Business Innovation and Incubation Center and the Innovation Hub, MG University is now moving forward to have translational research in a more effective way. The Attract Life program is an integrated research translation commercialization program at CCAMP, which is focusing to foster innovations for society by leveraging the strength of Indian academic research and CCAM's translational resources. This call will be open to all academic discoveries and inventions which require support for translation into technologies with a potential for significant impact in the different fields of life sciences, including areas such as human healthcare, animal healthcare, primary and secondary agriculture, and environment. The Sri Ramakrishna Paramahamsa Research Grant for Translational Biomedical Research is having a major objective to provide bench to bedside funding to enable the benefits of biomedical research to reach larger sections of the population. So, this should be a prime objective of any researchers to have a potential translational research project. So I emphasize the fact that all our inventions or discoveries or design of our research activities should be to address the requirement of the lower economic strata of our society. So what are the gaps between basic research to translational research? There is a long way to go. Say, for example, when we start our ideas to develop something within the laboratory and then to go for a translational research, you will find lots of gap areas. For example, the first and foremost step of basic science and innovation, that is GAP 1, which is to improve productivity of clinical development, proper ethical approvals, regulatory reforms, and consider health economics in decision making. And when we come to the innovation and clinical trials, which we may identify as GAP 2, that is to translational research or translate research into product, develop new innovation models, bringing more intellectual property laws into practice, health business economics and health policies, which are essential component for translational research development. Coming to GAP 3, at the stage of clinical trials and approved innovations, you will find that appropriate dissemination, adaptability, adaptive approach, and open minded psychology of patients will become more crucial. And at the end, addressing gap 4, that is related to innovation products and patients, we need to have appropriate post marketing surveillance after approval of phase 3 of drug based on clinical research from the cohort or epidemiological study documentation, which will become essential. If you look at translational research perspectives in India in view of different stakeholders, that is, we have the funding agencies, which are very, very important. If you look at the different stakeholders involved in the translation research, funding is very, very important in order to facilitate all our research programs, which will become very, very challenging nowadays to have sufficient required fund for our implementation of our research ideas. We need to have we need to face a very tough competition at the national level or in the international level as well. And there is a good partnership between pharma and academia, which is again very, very important. And then all our innovations need to be patented before we go for marketing our products. And then we need to address the approval or we need to address the questions of the government and regulatory agencies in order to make our discovery as a product which is available to the society. In the field of drug development, we need to start with the molecular pathology and then to cell biology and then proteomics, genomics, and then bioinformatics. So lots of stages are involved which will address different aspects which I have listed under different subheadings. So they need to have such a very wide kind of investigations for a drug development in general. If you look at translational research in agriculture, which basically brings scientists and stakeholders together to develop research that will address environmental challenges. For example, in order to have a very high yield, we might be using certain kind of fertilizers. And these fertilizers, are they environmental friendly? That need to be addressed nowadays. In the ester years, we had the green revolution based on the heavy usage of fertilizers. Now it has affected many of our water bodies, soil systems, so on and so forth. In the entire ecosystem got affected because of the extensive usage of use of such fertilizers. So the translational component research in agriculture need to be addressed in a very different way. That is, the partner's flexibility to overcome another constraint can increase the success of translational research, but it may harm the environment or the mother earth on a larger perspective. So that has to be considered very well in any research field. In the field of translational nutrition, which is basically a multidisciplinary endeavor involving nutrition science, food science, health science, these are all acting together. So the parallels between the rapidly emerging fields of translational medicine and translational nutrition are numerous, and their successes are complementary, as I have depicted here. Like, you have the nutrition knowledge which we have gathered through our scientific fields and which we need to translate to the society, which in terms will be addressing the nutritional solutions for health and wellness of the society by and large. And we need to fill the gap in the nutrition knowledge and priorities for future research. So this component is very important in all translational research, not only with the translational nutrition, but with other translational research areas as well. Energy is a big concern for us. All of us are worried about the next energy consumption of the entire global population. People are working on different aspects, like people are working on fuel cells, people are working on batteries, people are working on other alternative energy sources like solar energy, so on and so forth. So the major component in this case is to address the carbon level or the decarbonization of the global energy supply, which generally demand clean sources of hydrogen to use as reducing equivalents for fuel and electricity. So the next World War is going to be for water. That is the prediction given by many of the social scientists. So the next World War is going to be for water. Hello, am I audible?
Yes, sir.
Yeah, so there is an electricity, there is a power problem now, energy problem right now. Okay. So we are trying to have novel approaches to have, uh, efficient energy resources, such as fuel cells. So our group is also involved in developing, certain electrode materials for fuel cells and batteries, including lithium-ion batteries, working currently on water splitting to produce hydrogen without any chemical agents, and thereby to have a hybrid hydrogen fuel cells uh, in the future. So translation research for energy and beyond is very promising, but at the same time very challenging. The next World War is going to be for water. That is the predictions given by many of the social scientists. And all our water bodies are polluted to the larger level, I mean, which we cannot even imagine. And the reason pollutant in most of the water bodies are antibiotics. So these are due to various reasons, and we need to have a well defined policy to address water issues, waste management, so on and so forth. So water, sanitation, and hygiene development efforts have aimed to reduce morbidity and mortality and improve service delivery, and thus associated research has a strong orientation toward applied studies. So, for example, you have the pollutants from industry and the human generated pollutants in water bodies and soil, and the wastewater, which is need to be sustainable. So we need to have a novel approaches for sustained water treatment technologies addressing the circular economy. So this is a new terminology coined in the context of translation research to have a circular economy. Currently, our group is involved with the Swedish counterparts in Stockholm University to have decentralized water systems using a sustainable approach. So we propose to have active black carbon or activated carbon to have portable water systems for rural India. So this is an ongoing program. We are aiming at a very large EU funding for the same. And very recently, machine learning and artificial intelligence have become an integral part of our research activities. We are also currently making use of machine learning process to map the different spectral data to understand an in-depth knowledge or an in-depth understanding of the kind of molecules or ions involved in different type of materials during the process. So applied artificial intelligence and machine learning in translational medicine is also very, very important nowadays. For example, you have the raw data and you have the clinical data, and then you have different variables and different type of patients as well. For the same disease, every patient will be different. So you have a quite large number of data need to be analyzed, and then you need to have the feature extraction from this data, making use of certain algorithms, and then you can group them or cluster the patients, and then you can think of the kind of drug which could be administered on the patients, and then the predicting the patient outcomes making use of artificial intelligence and machine learning. So this will actually reduce the number of trial experiments with humans by and large. So the role of artificial intelligence and machine learning will become more prominent in the coming years to come. When we look at the applied artificial intelligence and machine learning in translational medical imaging, this is another field for medical personnel to analyze the data. So we are now trying to do the machine learning algorithms to understand the spectral data. The same way we can also make use of the medical imaging data. So currently we are involved with a big project with the Nanyang Technological University under the Spark scheme, developing novel magnetic nanoparticles for magnetic resonance imaging. So this is an ongoing activity with Nanyang Technological Medical School. So you have the source row, that is the image data, and then you can process it through the machine learning algorithms, and then you can make use of it for the translational research to have a better understanding of the medical images we receive through different techniques. In all these translational research, we need to have the industrial partnerships, which could be in terms of startup incubators, making use of the best human resource management, and then we work as a team. So that is what Mahatma Gandhi University is currently envisaging in the campus. We have the student startups through the Business Innovation and Incubation Center. And then we have the innovation hub they will provide. That particular center will provide the basic infrastructure facilities for the translational research. So this is very, very important. Most of the time, the industries, they prefer to have the already existing technological know-how for their product development. Only very few industries are actively involved in research and development for their translational research. And therefore, currently, when we look at the major issues like food security issues, energy security issues, water security issues, health security issues, which will require a very immediate kind of translational research to have product available in the market. If you look at the overview of the role of industrial collaboration for translation research, you can pinpoint quite good number of points for that matter. For example, the industry input into translation was seen as an important by all academics. There is no question about that. Every academic researcher would like to have an industrial partner, but it has become very, very challenging. As I have mentioned, already the existing industries, they would like to have the technical know-how, existing technical know-how for the implementation or the translation of their products. And therefore, the role of industries have become so crucial for the development of new devices at the translational level. And therefore, funding has also become very, very important. For example, we may require to have industrial partnerships for non-clinical research aspects. and then we need to have industrial funding for clinical research aspects. So you have the industrial partnership for non-clinical work, clinical work, and therefore the translational research part has become more complex for that matter. So the funding we receive from the government organizations like Department of Biotechnology or ICMR or Department of Science and Technology, Nanomission, there are different agencies. But at the same time, the funding is very limited. It will be for a short period. And the outcome we anticipate will take more time. And then who is going to fund the rest of the expenses. So that has become another important challenge or hurdle in the process of translational research. Okay. So industrial partnership is very important, whether it is related to biomedical or it is related to the energy sector or the water sector, whatever it is, we need to have industrial partners. So we need to look at the various assets both academia and industry bring to collaboration with the goal of translations. For example, industrial contribution to translation could be in terms of in the field of medical science, could be the compounds, data about drug compounds, regulatory knowledge, prior experience of translation, financial resources, processing capacity. The academic contribution to translation will be essentially on the context of extensive subject specific expertise, unique experimental tools, example animal models, which is again we can make use of the current simulation techniques so as to reduce the number of animals involved in such kind of experiments. And then to the translational level, it comes to the patients and then regulational value. So it involves these are all interconnected. Like the industry academia partnership is very important to have the translational research. Coming to the key cultural and institutional factors at the academic culture part, you have the data sharing which need to be open, the time which we may consider as unlimited, but it need to be limited to certain extent. Money, of course, is always limited. And the institutional features, if you look at patenting process, it may take time, collaboration support, the contracting process. Most of the time in the project, an individual principal investigator is involved. If it is an industrial partner, the industrial partner either could be a principal investigator or a co-principal investigator. If you have an industrial, a foreign partner, the foreign partner could be PI from the foreign side, and there could also be a co-PI. So the number of personnel involved in such kind of projects will become large, and at the same time we need to address the intellectual property right. So we need to have an agreement before we sign a particular project contract. From the industry side, we have the data sharing, which is normally not very open. Time is again limited. Money probably could be unlimited depending upon the company with which we interact. So the institutional features for the industrial partner, like the rate of turnover of personnel, company size or level of hierarchy, the structural changes. It could be either an individual project or it could be in partnership with an academia researcher. So this is also not a very flexible kind of things, because the industry will have its own targets to meet. Academia person, they fix the target, but to achieve the target they may take more time and with the constraint of money. So there are conflicting interests which may develop in between, because the industrial partner, they may like to have the results in a very short span of time, and in the academia the context, it may take more time, and therefore there could be some kind of conflict which may arise in the translational process. So how to bridge the gap between academia and industry? You need to set realistic goals. We may have different kind of ideas. You may have, as the Nobel said, like you may have thousands of ideas, but couple of ideas if you can realize, that is really sufficient enough. So we need to set very realistic goals, encourage industry interactions for students, and then you can also think of creating a strong alumni network. So some of our alumnus might be working in different industries. We can bring them. All the alumni could be brought to the campus and give an opportunity for the students to interact with them. So the industry interaction with students is very important. And what we can do is that we can send the students to different industries so that they will get a flavor how this translational research is being carried out at the industry level. You can also offer student-centric education, not faculty-centric education, so that we can provide the students with a practice-based learning to try and train methodological competencies. And we need to avoid rote learning and emphasize, that is, read, write, and reproduce. That concept we need to change, which should be more problem solving kind of approaches so that they can have real models which will be really helpful at the translational level. We need to blend both hard and soft skills judiciously, impart soft skills training, which has become now a very important component in most of the educational curricula. Use the concept of learning factories, like combine different approaches and existing concepts with emerging topics of the industry. Ensure smooth communication and coordination between academia and industry. That is, we need to invite industrial personnels to the campus very often. And this is happening in most of the engineering institutions. I think probably in IITs and all, it is happening very often, but in universities in general, which is very, very less. There are only very few universities which have industrial personnels in their campus. And we need to ensure now every research we carry out should have kind of a corporate approach. Like we need to have some kind of deadlines. We need to evaluate the progress very often, not at the end of the year, but in the process. For example, in my group now, I have started evaluating the progress of the students every week. We ask them to submit the reports, the work they have carried out in every week, and then what they are going to do in the next week, so that there will be a critical analysis of the progress, whether we are able to achieve the targets what we have envisaged to complete within a stipulated time period. A corporate kind of approach is very much now needed in all the research pursuits. The reason is that money involved is quite high and we need to deliver the output. That is, we need to go through the translational part and then to have a product at the end. We can also invite educators to the industry to understand expectations and recruiters to visit campus to empathize with the challenges of the industry. Which can be offered for a short period. So teamwork is again another important key factor in industrial collaboration. So we need to have very effective collaboration which can improve your team works together and solve the problems. So this is very, very important. If you look at the product development, I was visiting one of the industries in Sweden during my last visit a couple of months back, as I was invited to work on the India-Swedish program to develop, as I have mentioned, in the field of decentralized water system, rural India. We were invited to an industry where the partners they sit together because development involves different stages and it requires improvement or improvisation every stage, after the first product. So a team can only address such kind of things. So this is very, very important. Whether you work with industry or you work within a group of researchers addressing certain kind of issues to develop solutions, we need to have that kind of teamwork. So within the academia, within the industry, we need to have a good teamwork, and the industry academia partnerships, we should have a very strong teamwork. So this basically lead to a certain kind of startup incubators in which the students or whoever may be the the starters or the startup developers, they will come up with an idea. They require a kind of support, and then we need to have a kind of brainstorm discussions in order to address different issues. And then we try to find out what are the potentials involved in this, and we have developed the knowledge and the skill, and to develop and then to have the product development at the end. So this is a kind of activities we need to have, which cannot be prolonged without I mean, prolonged for a longer time period. So the technology business incubators are very much needed, like the startups, what would we need. So during my visit to US next week under the Fulbright scheme, I will be basically discussing with the different educational partners in the US to understand how the business incubations are being carried out, or the business incubation centers are implemented in US institutions and promoting student startups. So this is a very important aspect. NG University is also moving towards that. We have already started business incubation centers, so we can have the entrepreneurs to have a product at the end, or we can promote the entrepreneurship skills among the students to have their own startups. So we need to have the incubation centers, which also require certain kind of mentors among the academicians, a high level of research and education and seed funding, which is again an important factor. So the university is now providing certain seed money for the students to have the startups. So this is a new scheme, that is the International Center for Innovation Technology Transfer Entrepreneurship Technology Business Incubator, started by DSD, Government of India, which is a kind of support given to startups for early stage prototype intellectual property assistance. This is generally to promote a spin-off ventures involving entrepreneurs and in residence, that is small scale industries could be promoted through this scheme. So anybody can browse the site and look into the requirements. We also have started biotech parks and incubators over a period of time. For example, the Department of Biotechnology so far has supported 9 biotechnology parks in various states, like Biotech Park in Lucknow, Biotechnology Incubation Center in Hyderabad, a TIDCO Center for Life Sciences Bench Park in Biotech Park in Chennai, the Golden Jubilee Biotech Park for Women in Chennai, a Biotechnology Incubation Center Cochin. So this is again very encouraging information for us to explore the possibilities. The top 10 startup incubation centers in India, if you ask me, like IIM Mumbai, they have a startup there, NSCL program, and then the People First Incubation Center. And Kerala has also have the Kerala Startup Mission. They are promoting quite large number of projects nowadays through certain evaluation process for funding. So we can all explore these kind of things. And who are the agencies? They generally regulate the translation research. So we have the national regulatory bodies for translation research. For example, the Central Drug Standard Control Organization under the Directorate General of Health Services, Ministry of Health and Family Welfare of Government of India, and then ICMR, that is the Indian Council of Medical Research. They also have regulatory bodies to control or to have a high level interaction with the translational research ideas. So altogether, as I have mentioned, the key documents in translational research, whether it is related to health, energy, food, or water, or whatever it is, we need to have certain kind of funding, and we need to follow ethical guidelines of the Indian Council of Medical Research if you are involved in a high-level medical research, and then Indian good clinical practice guidelines we need to follow, and we can all have a look at all those things so that you will have a clear idea what we are supposed to do, what we are not supposed to do. And as I have mentioned, funding is very, very important. Funding is very, very important. Whether it is a support from the industry or whether it is a support from a government agency, that the proposal should be of very high level in order to have a very successful proposal. The grant writing is, or the grant proposal writing for translation research is very, 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 very important. So what are the important aspects you need to look into when we write a proposal? The grant proposal writing for translational research has different stages. We need to have, first and foremost thing is that, do we have a clear background of research for this particular proposal? You need to take plenty of time to look into that aspect. What are my strengths with related to a particular area? We need to be very clear and get criticism ahead of time. We need to explain the impact. We have to pick up the best group for the work. That is teamwork always the best. And then budget the project or the proposal very carefully and provide value for money. We need to give justification for each and every head of money we are asking for, and give a very clear methodology for the implementation. So these are the main factors which you need to bear in your mind before we submit a proposal. The common sections of a grant proposals involve like a cover letter, executive summary, the need statement, like what do you want to perform this research or this particular project. Goals and objectives should be very, very, very crisp. Methods, strategies, or program design should be very, very clear. Evaluation section, other funding or sustainability we need to bring in, information about your organization and the project budget. So these are the common sections and the gap areas we need to mention. In most of the proposals, we need to project the gap areas at the national level and international level. Whether it is in a national grant or international grant, we need to do that. So what are the basic characteristics of a good proposal? It should be very precise, very short, clear. You need to use very good language. Before you submit, you need to have the spell check and grammar check before you submit the proposal. Whenever you see a red underline, think about that, why there is a red underline with a particular sentence or a particular word, and then you have to rectify that. So you need to go through the proposal many times and make it very perfect. And writing the proposal of a research work in the present era is challenging task because it is a constantly evolving trends in the qualitative research design and the need to incorporate medical advances into the methodology. For example, I have formulated a proposal 6 months back, which I cannot directly submit for a call, which got a deadline next week, because I need to update the kind of references and what is the current status of the particular proposal now that need to be projected. So an old proposal we can always reuse with lots of improvisation. So writing a proposal is very, very, very challenging, and you need to have the inputs from time to time to have a very clear, precise research proposal. So 5 common mistakes in writing a translational research grant proposal will be, we will be too ambitious, like our goals will be endless. We need to have very few goals which we can address within a stipulated time period. So we need not be too ambitious about the research goals. Don't put so many goals. We have to put the goals which are doable. We need not to put very complicated kind of explanations. Everything should be very crisp, very understandable. A proposal should not have quite good number of pages, and I mean nobody is going to read the entire thing. If it is bulleted kind of targets, explanations, then it is easy for the evaluators to go through that. And most of the time we have a very inadequate academic level. So our basic understanding should be improved from time to time, and lack of experts in your team. When we induct a team or when we try to formulate a team, like I might be having certain kind of expertise, but I may not be having certain expertise which is required for the success of the project. In that case, you induct a personnel with that expertise and build up a team. And the plan should be very, very precise. So the five common mistakes generally people commit are too ambitious research goals, too complicated explanations, inadequacy to your academic level, lack of experts in the team, and no precise plans for the proposal. So main reasons for research grant proposal rejections are the deadline for submissions was not met. Maybe we'll finish it. Today is the deadline, 5 o'clock. We may finish it by 5 o'clock now, and we may not be able to submit that. So we have to get ready prior to the deadline. The proposal topic was not appropriate to the funding agency to which it was submitted. So most of the time, the funding agencies will have certain thrust areas, and when we formulate the headline or the topic, we need to have those kind of required thrust areas or themes in the uh, headline or the topic. Guidelines for proposal content, format, and or length were not followed exactly. The proposed question design and method were completely traditional, with nothing that could strike a reviewer as unusual, intriguing, or clever. Clever. The proposed area of study was not an agency priority for this year. So when we submit a proposal, our proposal should meet the theme areas of that particular agency's call. The proposal was not absolutely clear in describing one or more elements of the study. So the list I have given, the proposed method of study was unsuited to the proposed purpose of research. So the grant cycle is something like this. You have to generate an idea. We need to find the proper funding source for the same and develop our proposal. That is in a very systematic way. We have to do that. As I said, nowadays, unless until we have the industrial partners, our proposal will not be considered at all. Submit your proposal well in time and then manage your award. Like you need to have a very good understanding about the proposal, because when you are called for the presentation, there will be hundreds of questions from the panel. And then at the end, if your proposal is successful and over a period of time, you will be developing certain kind of solutions for certain problems, and that you need to share to the international community or the research community in terms of publications. And before you publish, try to look into the possibilities to have a patent. So you patent it and then publish, and then you can go for the translational research. So these are some of the areas of our research group, like we work on laser matter interactions to have novel materials and understand the basic sciences, and then we go for the translational work. For example, we are developing a search-based platform for sensing applications. We make use of laser matter interactions to a larger extent. Tissue engineering, we are very strong thanks to Pushpagiri Research Center in associating with us or giving us the opportunity to do the animal models. Over a period of time, many researchers have helped us in getting good publications, and some of the works are actually at the translation level now, clinical trial 1. And we also work on spintronics, like electronics. The next generation of data storage will be based on spintronics, multiferroic materials, optoelectronic applications. For example, we are working on solar energy materials for solar cell applications, batteries. High energy is another area we are currently working on. For energy, water purifications, we are making use of polymer nanocomposites, activated carbon, so on and so forth. We are also now into modeling and simulation, making use of high performance computing facilities and machine learning, as I have mentioned. So we work on an interdisciplinary aspect. Over a period of time, we have developed, or we were able to establish quite good number of experimental facilities at Mahatma Gandhi University, mostly through national fundings from different agencies. International collaborations, we have quite good number of collaborations across the globe with the different institutions. We also have industrial partnerships, thanks to Professor Sabu Thomas for his all initiatives to have international networking. He associates with DuPont, Apollo, General Cables, of Street Reliance, MRF, so on and so forth. And these are some of our international collaborations across the globe. We collaborate with most of the countries in the world. And this is a typical roadmap for IIU CNN. And we have already had the incubation center now. we have started working on fabrication of certain kind of devices, particularly the nano energy generators based on piezoelectric polymers. And probably by 2025, we will have the pilot scale up of nano products and commercialization of nano products by 2027. So you need to have a very clear cut roadmap for all our activities, some kind of year based planning and evaluation and progress evaluation at proper stages. So lots of institutions collaborate. I'm happy to mention Pushpagiri Research Center, Tiruvalla, for all our biomedical research, particularly for animal models with regard to tissue engineering. And lots of people, researchers collaborate with our group. Thanks to Professor Sabu Thomas for being a good mentor and a good partner for all our research activities. And we have done quite we have received quite good number of research projects from various agencies, and the list is not complete, but I would like to mention at least these people. They are part and parcel of all our research activities. And I'm very thankful and grateful to all my past and present co-workers at International and Inter-University Center for Nanoscience and Nanotechnology and School of Pure and Applied Physics. And without their hard work and then dedication, we were not able to achieve this much within a short span of period. And I am the PI for 4 Spark projects funded by Ministry of Human Resource Development, Government of India, which was started in 2019, but we got an extension up to 2022 because of the COVID-19. And these are the partners, international partners associated with that. We work on diversified topics on cross-disciplinary and interdisciplinary aspects. And these are the agencies, they have funded us enormously, like DST, Department of Science and Technology through Nanomission, Board of Research in Nuclear Sciences, DAE, for plasma imaging facility, UGC Government of India through the innovative scheme for the laser facility, Kerala State Council for Science, Technology and Environment, Department of Information Technology, and other agencies like UGC, DAE, CSR, Kolkata Center, they have funded. These are some of the research grants ongoing. Myself as the principal investigator and principal co-investigator. And these are some of the patents granted very recently. And we also have the international ongoing projects. The recent one is the India-Serbia project to work on multiferroid perovskite-based nanostructures for EMI shielding and photovoltaic applications. And with the Institute of John Lamour, the recent IRP grant. And then I'm the principal co-investigator with the ILM, another fund. CEFIPRA, this is again an Indo-French scheme granted to me as PI. And this is the BRICS project we have received very recently with Russia, China, and India. That is fabrication of three-dimensional nanocellulose-based multifunctional materials for tissue engineering and regenerative medicine applications. And a couple of projects, international projects, we have already completed. And this is the recent international cooperation work in the area of water purification or water security issues we are working on. In the initial grant we have received to have the kind of interaction meetings and all. Now we are planning for a very big EU grant for the same topic. So these are some of our patents. So I'm thankful to my team. Lots of people are involved. These are the present co-workers working on different schemes. Srijis is leaving for France very shortly, and Marin was in Ariel University, Israel. Shilpa is already now in Israel. Fadila is now in US. Badra and Bosley will be leaving for NTU Singapore very shortly. Anu is our time engineer. And Diba Sridev is working on machine learning for spectral image analysis. Sita Lakshmi is our very aggressive computational physicist, publishing quite a good number of papers within a short span of time. Nidish is working on laser matter interactions. Rahul has submitted his thesis very shortly. Satyanathan was our CIFIPRA PDF. Now he is moved to Korea, and we have inducted a new postdoc. Chitralega, I did not have her photograph now. Raji is working on the BRICS proposal. I mean, she is also a partner of the same. So these are our group members. Thank you very much. So let us have a very sustainable Mother Earth in the future for all of us to live comfortably. So translational research has become very, very important. Any research what we do in the laboratory should have a translational component, and that will help us to build up our society a better world. Thank you very much. I take this opportunity to thank Reverend Father Maureen Cheryl and Dr. Nebu for inviting me so cordially to this platform. And thank you all for your patient listening.
Thank you, Dr. Nandakumar, for that wonderful lecture. You kept us all glued to the chair for the past 1 hour. It was so interesting. You had a comprehensive description, and I'm sure the audience would have learned a lot. It's actually described all about the possible sources of government support. He has also given us a descriptive knowledge about how to elicit industrial support, and also has given us valuable tips and tricks as to what one should keep in mind when you're thinking about translation research, the government policies. All these are very much essential and imperative and important for anyone who takes up this research. And I'm very envious about the team, Doctor. You have a very wonderful team working along with, very
Yes,
efficient,
yes.
no doubt, under your leaderhood, they're all doing good. Thank you for
Thank
the
you.
talk, and it's now open for discussion. We don't have any queries in the chat box. If anyone wants to ask something or get some information from this learned person, you can put in the chat box, or you can open up the mic and speak.
You can also write to my mail, nkkalarical@mgu.ac.in. And whoever is willing to visit Mahatma Gandhi University, please let us know. We will arrange a lab tour. We have quite good number of facilities available, which is at par with any top institutions in the country. And we are also one of the best in the country. I'm very proud to mention that. And you can make use of these facilities. And all the facilities what we have established at MGU are open to all, and some you need to pay, but the facilities are available, and some are totally free.
Thank you, thank you. That is indeed very sweet of you to give us a welcome to your institute. And I hope those researchers who are interested will pay a visit to this particular esteemed institution and learn how things are being done. I'm sure it's going to be beneficial to each one of you who's going to take up research matters seriously.
Most welcome.
And sir, this is not a query. This is Dr. George Varghese.
Nice
Yeah.
meeting you, doctor.
Yeah. So good morning. Good afternoon, sir.
Yeah, good
And
afternoon.
we are actually in the dark, how to submit a research grant, because
Yes.
and you have given the clear steps how to do it, and what are the pitfalls and what are the mistakes we are likely to come across, because our research grant usually we give up to ICMR or maximum that university level or Kerala government level, But now you have given us the idea how you can even go up to the DST. I'm telling about Pushpayari Dental College. I mean, medical college, they are going a little more higher up, the PRC and all. So it's very good that you have given us the outline and where are the areas where we can search for by getting the grant. Thank you very much, sir. Thank you very much, and have
Most
a
welcome.
safe journey to Calcutta.
Yeah, most welcome. Thank you very much. Yes.
Well, then shall we move on to the next session? I would transfer the mic to Dr. Soumya, who's in charge of the proceedings. And thank you for giving this opportunity to moderate this session. Thank you so much.
Thank you all. Bye.
Thank you, Nandakumar sir, and thank you, Vikram sir, for the sessions, for the leading the session and for the moderating. Now we will be taking a break for lunch, and we please join in by 1:15, 1:30. We hope to start with Dr. T.V. Kumar's Anil Kumar sir's lecture. Right. Thank you. Good afternoon, sir. Professor TV Anil Kumar, sir. Sir, welcome once again to the workshop. We will start at 1:30. Sir, shall we start? TV Anil Kumar, sir. Good afternoon once again to our workshop on translational research in tissue engineering. We begin our afternoon sessions now. I invite Dr. Elizabeth Joseph, Professor and Head of Department of Periodontics, Pushpuri College of Dental Sciences, as moderator for the next session.
Uh, Thank you, Team BioRadiance 2022, for this very relevant topic of translational research in tissue engineering. I'm happy to introduce Dr. T.V. Anil Kumar to you. Dr. T.V. Anil Kumar is the scientist and the head of division of experimental pathology of the Biomedical Technology wing of Sri Chitra Institute of Medical Sciences and Technology, which is in Trivandrum in Kerala. And he will be talking to you about a very relevant topic. It's how to begin a career in translational research. Dr. Anil will be covering a stepwise guide to beginning a career and the various challenges and attractions of career in translational research in tissue engineering. Welcome, Dr. Anil Kumar. The stage is yours. Doctor, can you hear us? Dr. Anil Kumar? Dr. Anil Kumar, can you hear?
ഹലോ മിസ്റ്റർ പറയുന്നൊന്നും കേൾക്കുന്നില്ല സ്ക്രീൻ കാണുന്നുണ്ട് ഫോണിൽ കേൾക്കുന്നുണ്ട് മറ്റേ ഓക്കെ
Yes. You are audible. Little bit louder.
Yeah, is the screen visible?
Yes, screen is visible.
Yeah, okay. So, can I start?
Yes, sir. You can start.
Yeah. Thank you, madam, for the kind words of introduction. Hope I am audible now.
Yes, sir. You're audible.
Yeah. Okay. Okay. So the research, I mean, topic is assigned to me is, how to begin a career in translational research. Probably I am going to end my career in translational research. So, and my research is, or my career is centered around some topic called as comparative medicine. And my background is, I'm a veterinary doctor basically. I believe most of the audience here, they are dental doctors. So there is a common thing amongst us is, we are all clinicians. And probably I have a PhD title. That's why I became a researcher, and I'm also a pathologist. And I have some expertise in lab animal science, uh, and also a specialty called toxicological pathology. So I talked all these things not to impress upon about my credentials, but rather this is a blend of so many topics uh, in which I happen to be in. And I have also happen to be in some of the finest institutes around the world, learning about research and doing a lot of research activities. The first one where I was first institute where I was associated with was Royal Postgraduate Medical School in England, where I was mostly doing fundamental research, and that was on apoptosis and things like that. But today's topic is nothing to do with those type of research. But as far as the translational research is concerned, these are the main topics which I am going to cover through. First thing is, get yourself educated, specialization and super specialization, which I already told you about my background, and lateral specialization in management, quality control, intellectual property rights. All these things would certainly help us. Some occult skills related to computer communication, public relations, etc., could be useful. And also, you have to identify your passion for research, be it fundamental, applied, or clinical, or translational. If you can manage all those things, that is well and good. But as I said earlier, I was mostly doing fundamental research in beginning of my career, but later on I came to translational research, applied research, and gradually to translational research, and a little bit of clinical side. And managing source of funding is important. Managing publications and patents is important, and technology management is also important when you talk about.
Hello, sir. Have you started screen sharing?
Yeah.
No, the screen is not visible as yet.
How do I? Let me hang on. There is no screen share option. That may... I can't see any screen share option allowed in this.
It's an icon with an arrow. It's here at the bottom of the screen.
Let me.
At the extreme bottom of the screen, there's an icon with an arrow pointing up next to the hand. Yes, yes, sir.
Yeah.
Yes.
Sorry, I was talking about my credentials here. I was talking about my multiple backgrounds apart from veterinary science, being a pathologist, and also being a researcher. So I'll be talking more about managing a career in translational research rather than talking about how to begin a career in translational research. Yeah. So the key components which I was going to talk is, I mean, they are listed here. But at the interest of time, probably I will skip this slide for the time being. I'll be dealing with some of these topics, managing funding resources, uh, identifying area of research, etc. So I was talking about my earlier career research. I was doing fundamental research in Royal Postgraduate Medical School in London, and then I was doing a little bit of clinical research related to veterinary clinical practice, diagnosing these type of X-ray photographs, histopathology photographs, all those things will be there in this type of research. But when I joined Chitra Institute about 20 years back, the topic which was assigned to me was evaluation of biomedical devices and biomaterials, and that is in fact translational research. So I was doing this for a few years, for about 3 or 4 years. Then I thought I must do some mainstream research also. So with this objective, I went to Ireland, National Center for Biomedical Engineering Sciences, and there I met this gentleman. His name is Abhay Pandit. He is probably same as equal age as mine. So he asked me, why can't you do some research, something like what this gentleman here at the top. His name is Stephen Badilak. He was working on mammalian-derived scaffolds. He is a medical doctor, he is a veterinary doctor, and he is also a PhD doctor, multiple specialty. So what he was trying to do is, with his veterinary background, he isolated extracellular matrix from small intestine of pig and then used it for regenerative medicine or tissue engineering applications. So Dr. Abhay Pandit asked me, why can't you do something similar to that? Probably because my background was veterinary science, and I was trying to find out a suitable organ from which extracellular matrix can be isolated. In fact, when I went to literature, almost all organs have been studied, and extracellular matrix from most of the organs have been isolated, recovered, and were used for tissue engineering applications, except one organ, which is gallbladder. So I decided to select porcine pig gallbladder for my studies. And from this pig, I mean, gallbladder can be collected at slaughter, and there are so many reasons why gallbladder became suitable for me. And this is how you convert your intellectual thinking or research, how we develop our research skills. First thing, nobody has used gallbladder for research. Second thing, I found all reasons why this will be a very ideal or suitable organ for tissue engineering scaffold preparation, because it's a fibromuscular organ. Unlike small intestine, it is not contaminated with blood or with microbial organisms. So recovery is a relatively easy process. What I was doing when I came back from Ireland, I started looking for funding source. There are so many funding sources, and from my funding, these are the three major sources I got from Department of Biotechnology, Government of India, Chitra Institute, and of course student funding. You find that getting started is very, very important. First, funding is always a problem. First 2 or 3 years, I was searching for funds, and in 2008, I came up with the DBT funding, and then started more research activities. And then these are the steps involved in standard technique for preparing tissue engineering scaffolds from animal organs and tissues. And this will involve collection of the organ from slaughterhouse, then it will be put in some antimicrobial agent or tissue culture media, and then it is transferred to laboratory. Then a process called a decellularization is done, and for the decellularization, so many techniques are available. Treatment with alkali, acid, enzymes, so on and so forth. And then it will be finally treated with a decontaminating agent to decontaminate thoroughly, and then you go for treatment with a stabilizing agent, particularly a cross-linking agent like glutaraldehyde. And then you go for long-term storage or terminal sterilization, and long-term storage by freeze drying or radiation or ethylene oxide treatment. So when I thought about this, because of my pathology background, I know that as a cross-linking agent, aldehydes will do some tissue preservation, then my objective is not to use any cells from these organs. So I avoided most of these steps which are related to scaffold preparation, and I treated with the cross-linking agent simply straight away formaldehyde, and then it was brought to the laboratory. I avoided all the chemicals, but a mechanical procedure was done to separate out extracellular matrix from gallbladder, because gallbladder has the tissues are arranged in different layers, so it is easy to remove the mucosa and also the serosal layer, and the fibromuscular layer in between was the scaffold. So this is the innovation which has happened there. I avoided most of the steps involved in routine preparation of tissue engineering scaffold. I call it as non-detergent, non-enzymatic method, and this resulted in so many publications and then a patent also. And the Indian patent is still pending. So very, very simple procedure. Essentially, what has happened is the procedure avoided avoided most of the complicated steps which are routinely practiced by tissue engineering scaffold preparation specialists. So that is my objective is to find out whether this technique will be suitable for other hollow organs like small intestine or urinary bladder, and each one of them work very well, and Indian patent application has been submitted for all these things. Once you do this, the first step in biomaterial preparation is you have to study biomaterial properties. Just a couple of examples here: histology picture, scanning electron microscopy, energy dispersion spectroscopy, FTIR, and so on and so forth. There are hundreds of procedures by which you can characterize a biomaterial you have isolated, and out of all these things, this particular procedure, what we call as identifying the number of cellular residue in the material from animal tissues, that is very, very important, because if cellular remnants are there, that can cause graft rejection when it is used as tissue engineering scaffold on the body. And for this very, very simple technique, you can do a histology preparation of each of the scaffold, and then count the number of nuclei. And here you can see that the gallbladder scaffold compared to the commercially available scaffold, the number of nuclei are very less. And you can count these nuclei, and then you can see that gallbladder scaffold is having very, very few number of nuclei. And you can also do a DNA estimate and also collagen estimate, elastin, galactosaminoglycan estimate. All these things in the scaffold can be done, and you can say that your scaffold is as good as or better than what is available in the commercial market. The most important point here, the process of decellularization need not result in so-called acellular scaffold, but some amount of cellular remnants is acceptable. And this is another important point where he is balancing applied research with the translational research. So far, I have been talking about applied research, but when we go for translational research, the safety of the material will have to be done. And incidentally, the biomaterial properties evaluation has many investigation procedures, and they overlap with safety evaluation procedures, or so-called preclinical safety evaluation test. So I did all these procedures, and then the results of these procedures gave me very good confidence, saying that this material is as good as a commercial product which is available in the market. And when we do this, it is preferable to do these things as per standard procedures or ISO protocol, and that will give lot more confidence in your test results, as well as courage to go for product preparation. And when you have to balance little bit of exercise between balancing fundamental research and applied research. In this picture here, so-called proteomic analysis was done to see the number of proteins present in the gallbladder preparation I have. It contains so many proteins, and out of all these proteins, the gallbladder scaffold contain certain proteins which are related to wound healing. So I was going through a little bit fast because of the interest of time. So wound healing molecules are present in this scaffold. So that gave an indication that this material can be used for preparing wound healing matrices. So straight away you go for animal experiment, you graft it with a rabbit model, you create a wound and then graft your material onto the wounds and see how best it is healing compared to products of similar origin. And this was awarded a prize in World Biomaterial Congress, which is the Olympiad of Biomaterial Specialists, and also some patent. Patent was also awarded. Use of this material for uh, wound healing application. And then any wound healing application, the objective is to speed up the process of wound healing. So this material, when it was tried, so many parameters are studied and the number of days taken for wound healing was calculated, and compared to unassisted healing, this material healed the wound much faster. And this was proved by histopathology and so many other investigation procedures. So in short, the gallbladder scaffold, which was isolated, it was having accelerated wound healing, and most importantly, it was also having it induced less scarring. So this material was identified as a wound healing matrix that will facilitate faster healing with minimal scarring. And so many parameters will have to be evaluated. And then this CDS, or the cholecystectomy derived scaffold, gallbladder scaffold, it was used for growing cells, and this was proved that it can be used for bioartificial skin preparation or even as a cardiac patch. So this type of research gives you a lot of translational potential to the material, and then you have to balance whatever you have learned in the laboratory to take it to clinics. Because if you can't take your research finding to the clinics, you can't do much. Here in this picture, this was a piece of research done in collaboration with the veterinary college, veterinary Manuti. They have used this material to as a wound healing patch for treating lacerated wounds in dogs, and it was showed that this material again favors faster healing. One more application was also tried to treat corneal ulcers. There also the CDS, or the cholecystectomy derived scaffold, was healing corneal ulcers in German pug. Then you have to find out, you have to tell others that your material is doing good, and then you have to prove how this mechanism of healing is happening. Probably these are the three important parameters, I would say. The cholecystectomy derived scaffold, it modulates angiogenesis, it modulates inflammation, and it also modulates collagen remodeling. So these were observed to do parallel research to support your translational potential. And here, the list of immunocompetent cells I studied in my lab, which was given as a topic given to a PhD student, and then so many cell types were detected by immunohistochemistry, and these cells were counted. In short, what has happened is the macrophage reaction, macrophage lymphocyte polarization was happening during healing process, and that was also proved. Again, it induced, the CDS induced favorable tissue modeling reaction rather than a pro-inflammatory graft rejection reaction. So this is a very, very significant finding which will support or enhance the translational potential. Of course, all these findings will have to be published and documented very well. With this confidence, we did a trial whether diabetic foot ulcer can be treated with this material. So we created a diabetic rat model was used, and then studied the angiogenic potential of this material in rat wounds created in diabetic rats. And then it was found that it facilitates angiogenesis, and then this was also published. A modified cholecystectomy derived scaffold was inducing angiogenesis and faster healing in diabetic wounds. So this can be used as a wound healing patch for diabetic ulcer. The next stage of the research was to convert this material into powder or as gel formulations. And this gel formulation, which is shown here, is was used to coat polypropylene meshes, which are used for uh, hernia repair graft. And when this material was coated onto routine polypropylene meshes, the healing potential was enhanced and the chances of graft rejection was reduced. And as if those of you who are familiar with the hernia repair, you'll find that many cases revision surgeries are required very frequently. So I believe that by coating this material, coating polypropylene with this gel formulation, it prevents graft rejection. One more topic was tried, whether it could be used for as a cardiac repair patch. There also we found that the cardiac repair is possible using this material. So in short, in summary, this extracellular matrix scaffold was prepared from porcine cholecyst, was prepared without decellularization and enzyme treatment. So the quality of the scaffold was very good, and the biomaterial properties of the scaffold uh, were comparable with the commercially available scaffolds, and it is less immunogenic, and then compared to similar scaffolds prepared from jejunum, small intestine, and urinary bladder. It can also be converted into powder and gel formulation, and so many animal models were used to demonstrate the healing potential of porcine cholecyst derived scaffold. And with this, so many PhDs were produced, publications were made, patents were applied, and then also got several appreciations and awards in several forms. World Biomaterial Congress, Indian Academy of Medical Sciences, Kerala Science Congress, etc. And with this, we went for a technology transfer, and technology transfer is very important. When you have to identify the technology, I mean, if you are very, very lucky, you'll get a technology partner, and then you will have to work hand in hand with the technology partner to develop a product. So, and this product was developed, what is called as Colidum was developed, and we had to help the industry partner uh, to set up their pilot production plant and also in quality control. So at the moment, I can say that the Drugs Controller General, which is Central Drugs Control Standardization Organization, they are supposed to give licenses for this product. First, they will give a manufacturing license for test purposes, and it was given some time ago, and the Union Minister of Health, uh, he was happy to do the technology transfer for me. And at the moment, I can say that last week I was told that the manufacturing process or for test purposes are over, and now they are going for complete license, what we call as manufacturing license for marketing, and I'm looking forward to a positive result in about a month. In short, although not very systematic, I was giving a brief talk about the process of translational research. You'll have to integrate fundamental, applied, and clinical research with hand in hand with the translational research. Funding has to be identified, establishing a laboratory infrastructure and basic necessary facilities will have to be established. And then, whenever you decide research topic for students, you give different pieces, but focused on a central theme. And your job as a investigator is to integrate all the research finding together, and managing publications is also important. And finally, if you are lucky, in my case I was very lucky to find out a technology industry partner, and you have to give support for industry partner for establishing their pilot production, scale up production, and interaction with statutory agencies, preparing their documents is very important. uh, Hope with a brief time I have shared my experience as a researcher in developing a product. So I'll be happy to take questions if time is permissible. Thank you very much. So this is my research team. Thank you.
Thank you, Dr. Anil Kumar, for the detailed insight into your research activities and how to integrate translational research into translational outputs. Does anybody wants to clear something, verify anything?
Sorry that I have to go a little bit faster because of shortage of time.
I think you have cleared most of where probably have verified most of what they wanted to know. So shall we move on to the next session? Thank you once more, Dr. Anil.
Okay, thank you, madam.
Thank you.
defect by another surgery so that you can see the defect. So this was the defect, and this has been obliterated completely by some form of tissue. If you want to know whether what type of tissue is this, you have to go for a histological section and you have to demonstrate cementum, alveolar bone, periodontal ligament within this tissue specimen. But this is not possible ethically because you cannot take out a section of the regenerated tissue from the live human mouth and subject it to histological examination. That is unethical. So ethically, a re-entry surgery and histological evaluation of this re-entry surgery is not possible. That is a limitation of evaluation of a regenerative procedure. Now, does periodontal regeneration really work? The data is, the available evidence support that all regenerative techniques can restore a fraction of original tissue volume. You cannot restore the periodontium to the pre-disease level most of the times. It is possible in very few situations, but evidence says that it is not possible all of the times. And apart from that, regeneration is not possible in all type of bony defects. You have to select the bony defect properly. Cementum formation or cementogenesis, that is the tissue that covers the root portion of the tooth, is essential. It should be demonstrated histologically to prove periodontal regeneration. And studies that claim regeneration do not use histological evaluation. So again, we are limited. We are in a, what do you call that, in a stagnant way to prove the outcome of our regenerative procedure. And finally, whether the statistical significance you get in the studies from the surrogate outcomes equate with the clinical significance. All these are areas of research that we have to contribute in the future to make this thing happen. And the real biological challenges in periodontal regeneration are, there are 4 types of tissues that are involved. It is a complex process. And the periodontal wound is exposed to microbial challenges of the oral cavity that again affect the healing process. And mobile structures are associated in the periodontal wound. Sometimes, sometimes the tooth that is involved may be mobile. So during the initial healing period, mobility should not be there. So we have to address that. And lot of host-related factors, patient-related factors also come into play during this regenerative process, during the healing period. These are the biological challenges, and the technical challenges in regeneration are the availability of an ideal material. We don't have an ideal material still, till date. And there is a lack of carrier device to deliver biological mediators into the defect. So that is an area of research. And we lack a comprehensive method to evaluate the outcome of the procedure. That is also one of our limitation or challenge. And finally, the cost involved is again another challenge. So these are the challenges we encounter in periodontal regeneration. A properly conducted translational research can answer few of the technical challenges that we address here. So to overcome these challenges, the skill, experience of the clinician, knowledge of the biology of periodontal wound healing, and appropriate patient and defect selection are essential. Apart from this, judicious application of the degenerative device and technique is very important. That is what I told in the beginning. The role of translational research in this area is very critical. Now, how can we incorporate translational research in regenerative dentistry? We can develop an interdisciplinary approach towards designing and evaluation of regenerative materials and systems. I know I did my post-graduation in Trivandrum when I did in way back in the late 1990s, and during that we have associated with Sri Chitra Tirunal Institute. My professor, Dr. Nandakumar, has done a lot of work on different biomaterials that is designed by Sri Chitra Tirunal Institute. So we have to incorporate the skill of not only the clinicians, researchers, basic researchers, the scientists, experts in biomaterial field, all these things. So there should be an integration and increase efforts to determine the specific cells required for regeneration of periodontium. We have to decide that. And importance of local factors for regeneration should be addressed. And this is an area that is delivery systems. Which system will properly deliver, carry the factor and deliver it into the site to bring about regeneration in appropriate way. Investigation of the influence of host and environmental factors on the tissue response is also important. Critical analysis of benefits derived from regenerative therapy compared to other treatment modalities on long-term tooth retention is also a factor. And presently, cell therapy or cryocytotherapeutic approaches have been investigated to overcome limitation of the existing procedures in periodontal regeneration. So stem cell-based therapy is a choice. And cell-based therapy in dental healthcare raises many biological, technical, and clinical challenges, as you all know. And there are a lot of regulatory issues the scientist or clinician working in this area may face, right from animal models, when they do something in the animal models, to human trials. So the entire cycle of translational research has got lot of stringent regulatory requirements to overcome. So that is an important area thing. And stem cell therapy has wide range of application in medical care, right from organ regeneration to treatment of organ failure. And the translational arm of stem cell science in dentistry remain in a relatively primitive state. Still, that is a problem. That is, and because of that, we have limited data on this area. But the collective progress in the stem cell technology, biomaterials, engineering, and molecular medicine has helped this area in a tremendous way. But still, what I feel when you look into the literature, we have there is a paucity of data, and we are still in a primitive stage. Let us see what are the problems associated with stem cell research and application in dentistry or in periodontal regeneration. One is there is lack of quality evidence because we don't have good quality evidence, strong evidence in this particular area because we are in the primitive stage of translational research or bench result, I should say, laboratory result. And the process is highly expensive, and it is usually thought that teeth are not essential for life. So it won't get that much priority what the other organs get. And the delivery of cells to the site is very difficult. In an intraoral defect is very difficult, and evaluation of intervention is difficult. Timing of treatment is also a concern, and safety of the patient always, as always, is always an issue in translational research. And these are the challenges and considerations in designing stem cell based translational research. One is cell procurement is not going to be easy. From where you get the cell, cell expansion or multiplication of the cells, that is a challenge. Delivery of the cell, there should be a suitable delivery system to deliver the cells, appropriate cells into the defect. Cell transplantation, outcome evaluation, and safety. So these are the certain challenges or areas that we need to address if we want to improve stem cell based research in dental scenario. There are concept of cell sheet engineering where specific cell types with signaling peptides are incorporated within the carrier construct. So that is a promising area where we need to have more research to develop an ideal construct which can deliver stem cells into the defect. So stem cell therapy, we need to have good understanding on stem cell biology and stem cell therapy, and it's a roadmap to clinical practice from bench to bedside. So cells collected are expanded, and it is stored or incorporated in various devices, and it is either injected or implanted into the defect to carry out the healing potential and to bring the desired outcome. So that is how the stem cell therapy, the cycle of stem cell therapy is going on. Now, a few words about health disparity, because my topic is to address health disparity in translational research in regenerative dentistry. I have just brushed through the different aspects of regenerative dentistry or periodontal regeneration, the challenges, the considerations, and the difficulties we encounter in periodontal regeneration. Now, health disparity is defined as the preventable differences in the burden of disease, injury, violence, or opportunities to achieve optimal health that are experienced by socially disadvantaged groups. Elimination of this disparity is the bottom line in achieving health equity, because everybody should get the equal health care, whatever be their social status, age, whatever be their demographic details, they should get. They are supposed to receive what you call equal health care facility or healthcare delivery. The health differences in population groups are mainly attributable, at least in part, to genetic, social, and demographic factors. And it adversely affects many communities in the world due to the influence of social determinants of health. The members from this socially disadvantaged group have carried the burden of historical trauma, discrimination, and marginalization, and have experienced worse health and greater health risk. So this should not happen in our future research. And these are the major health disparities. due to race, due to ethnicity, sexual identity, age, disability, socioeconomic status, and geographical location. Based on any of these, people may suffer health disparity. And these are the domains where the health disparity become visible. In mortality, the people who are sidelined or marginalized will have a high mortality rate. Life expectancy, burden of the disease or prevalence of the disease, mental health, uninsured or underinsured, lack of access to care. So these are the areas where the disparity become visible and obvious. And how can we address these disparities in translational research in the industry? We have to address these disparities, that is of no question. And is it we are not able to address this because of a lack of conceptual framework in translational research? We will see. There are certain techniques they mention in the articles to address these health disparities and to make the translational research outcome more amenable in a more equitable way to all sections of our population. One is, I told you this is a cycle. The need comes from the community, and based on that need, the laboratory trials are initiated, and the result of these laboratory trials are translated to a very controlled situation to assess the efficacy to bedside. And if the efficacy is proven good, it is taken to the community back. That is the effectiveness trials. That is how it goes. So we can do some adjustments in all these steps to eliminate the disparity. One is you have to identify the disparity and the root causes of this disparity. Assess public health needs. The real need of the public or community level factors should be assessed. And we have to target the needy based on the racial and ethnic minorities. And the use interdisciplinary collaboration to conduct research in uncontrolled conditions. And epidemiological assessment and community engagement are mandatory in avoiding health disparities. That means if you do an epidemiological assessment, you have to gather information pertaining to the social determinants of health. What are the determinants that result this particular health disparity? And we should identify the population disproportionately affected by a specific disease or condition. We have to identify that population, we have to assess their need, and as per we have to deliver the outcome of our translational research. And the community should be engaged in the research process. That is, we should have a member, because the include researcher with good rapport with the community. The people who are to receive the outcome of the research should not have a stranger phenomenon. They should feel like these people have come, is one among us, and they have come to improve our health standards with a new technique or with a new preventable method. For this, they mention the include researcher experience in community-based participatory research. So more of a sort of qualitative research approach we need to apply in this area to prevent the major disparity in health determinants to happen. So a community-based participatory research will promise better results, as they say. Now, from the bench, we should provide opportunity to the target population to provide input in the research process. This will help to avoid obstacles later in the later stages of research, because it should not be unfamiliar to them when the outcome comes to them, when the particular treatment modality comes to them. They should be familiar with that. So there should be a good interaction with the target population regarding the earlier stages of the research, during the earlier stage of the research, during the bench stage of the research, or during the bedside stage, that is the phase 2 trials of the research. A public-private partnership concept is good, as they say. And to the bedside, from a clinical research perspective, do practical clinical trials rather than traditional clinical trials. That means when you do a clinical trial, a particular clinical trial, if it is done properly, there are a lot of stringent criteria. That is possible in a controlled situation, in phase 2 trials. When you do it in a small number of patients, in a very controlled environment, when we apply that, it is possible. But when you go for the next level of trial, effectiveness trial, this may not be possible. You have to ease out certain stringent criteria to make it more amenable, more effective for the community. So for this, we have to do something. When you do the effectiveness trial, to make it more amenable to the community, more palatable to the community, we have to do something. That is why they say that do more practical clinical trials rather than stringent traditional clinical trials. So that's an area that we need to address. Now, after this, we have to put it back to the community. And when you put the treatment or the preventive method after the research, the end product of the research back into the community, we have to detect the barriers in treatment utilization. If the treatment is not utilized the community, the whole research process is in vain. So, what are the barriers, if any, that prevent them from utilizing the research, the treatment? That has to be assessed. And we have to do evaluation of interventions in some framework. They have given a REAM framework, that is the Reach, Efficacy, Effectiveness, Adoption, Implementation, Maintenance framework. There are a lot of framework available. Anyway, evaluation of the intervention in the community, the outcome in the community should be evaluated properly. And that there should be an ecological assessment where we should identify other determinants that is contributing to the disease before implementing the intervention, because a disease can be multifactorial. So all the factors that are contributing to the disease should be assessed, and we have to use technology in collection and analysis of large volumes of data. So this is what we should do once you put the outcome or put the treatment into the community. Now, I have gone through a very beautiful model which will address health disparity in translational research. If you follow this model, the majority of the disparity can be addressed, because this is the area of public health research. There should be an integration of public health research, biomedical, basic behavioral science research, and clinical research. So there should be an integration of all the three handles or arms of research together if you want to avoid health disparity in a translational research. There should be direct we should detect the disparities in the disease. The incidence and prevalence should be detected, and we should determine the underlying causes of the disparities, and we should treat the intervention or test the intervention with human subjects or populations. The T1 arm of the translation research and T2 arm of the translation research should be integrated in the proper way, along with integrating all the three arms, the public health research arm, the basic research arm, and the clinical research arm together. If done together in a cyclical manner, in a proper manner, health disparities can be avoided to a great extent, as they say. Now, let us see what is the present status of stem cell based therapy in periodontal research. We usually do preclinical animal models in small models, but evidence have stated that there is no evidence that is available regarding the outcome of a regenerative material in periodontal regenerative therapy on an ideal supracrustal alveolar bone defect. It is very difficult to create an ideal surgical defect. And another thing is cell delivery or administration modes, whether it is injection or matrix guided, that is not defined till date. And the biomaterials used for cell delivery, whether it is natural or synthetic, you have to define. And evaluation of the outcome should be, or evaluation of the intervention should be more defined. So with that, I am coming to my concluding remarks. In order to address health disparities or to avoid health disparities in translational research in regenerative dentistry, we should encourage and support the cycle of translational research. All arms of translational research should be encouraged at a government level, legal level, and institutional level. And we should integrate all components together in this particular process. We should identify the health disparities in a target population and identify the root cause of that health disparities. And we should take measures to correct the cause of those disparities. And we should include the concerns of the population and eligible candidates from the population in the research process. And finally, we should evaluate the outcome of a developed product and utilization of the product by the target population on community or the community. If you do that to a great extent, the disparity that is associated with translational research can be avoided. And these are my references. I had a few articles from New England Journal of Medicine. That's a very beautiful article. Then Indian Journal of Dental Research, I have got a good article. And the Journal of Cell Transplantation, this is a very beautiful journal. You can have a lot of information from this. And European Journal of Oral Science, I got a good article. And NIH, National Institute of Health Public Access article in 2008. These are my references. And I should acknowledge Dr. Jay Krishnan, former professor at HOD, Department of Community Medicine, GMC Kozhikode, Dr. Priya and Dr. Biju George, faculties of Department of SPM, Kozhikode, for their valuable inputs and guiding me in this presentation. And thank you for listening patiently. Thank you.
Dr. Harikumar, that was a detailed explanation of regenerative dentistry, more so in periodontology, and also your insight into health disparities and inequities in the community. Definitely, regenerative dentistry is very much in the clinical, I mean, little bit in clinical practice, and we need a lot of translational for that to get it into a full-fledged treatment modality. I think students, those who have, some of you are doing seminars and research in regenerative dentistry. Any doubts or any clarification? Thank you once more, Dr. Harikumar. We move on to the next section.
Thank you, Elizabeth ma'am, for moderating the session, and thank you, Harikumar sir, for your very informative talk. We now move on to the next session for the day. It is my pleasure to welcome Professor Dr. Anthony George, who is the professor and head of Department of Oral Pathology, MES Dental College, Perinthalmanna, to moderate the next session. Anthony sir, I hand over the session to you.
Thank you. Uh, Hope everybody having a good learning experience. And I'll thank the moderators for the giving me the opportunity for being a moderator for this Radiance Web Radiance 22. And if the participants have any questions, they may please type it into the chat box so we can take it up at a later time when the session is over. Other times, please keep your microphones mute. I think Sheena has her microphone on, so if you can mute it, so it will be good for the online experience. So it is my privilege and to call upon Dr. Betsy Joseph, Professor Department of Periodontics, Savita Dental College, Chennai. She is speaking on the current status of phototherapy in periodontal regeneration, which includes photobiomodulation, photodynamic therapy, and phototherapy in conjugation with nanotechnology. So I hand over the mic to Dr. Betsy Joseph for the next 30 minutes. Thank you.
Thank you, sir. At the very outset, I thank Pushpagiri Research Center and Department of Periodontics from Dental College for giving me this opportunity to make this presentation. I hope my screen is, I've done the screen sharing. I hope it's clear.
Yes, ma'am.
Yes.
Yes, please.
Thank you so much. So today, after the wonderful presentation by my senior, after the wonderful presentation by my senior, Dr. Harikumar, I'll be doing a very small presentation on how we can use the energy of light, the light energy, to enhance periodontal healing, wound healing, regeneration. So periodontal disease is very commonly seen in more than 5 to 15% of the population, in most of the population that you see, and there are different treatment modalities. And at the end of any type of treatment modality, you are aiming at getting regeneration. So there is non-surgical therapy and surgical therapy that is utilized. So in non-surgical therapy, along with local drug delivery, host modulation, antimicrobial therapy, there is another therapy that is used, the presence of light. So which we can call as phototherapy. So when we say phototherapy, probably this is the image that comes to us, because from time immemorial, light has been used for various types of treatments, to treat skin disease, to treat different eye diseases, neurological problems, even dental disorders. So when light is used for the treatment in the oral cavity, there can be two types of light that is used, non-thermal and thermal, and we'll be focusing on the non-thermal properties. So basically two treatment modalities, photo, biomodulation, and...
Sorry to interrupt. Ma'am, have you changed your slide? The slide isn't changing on the screen.
Yes, I have changed to non-thermal and thermal. Can you see it?
No, ma'am, the slide is not changing.
How about it now?
Yes, it is changing now.
Okay. So I'll just try to do a slideshow once more.
Yes, ma'am.
Is it now non-thermal and thermal?
Yes, it's non-thermal and thermal.
All right. Thanks for letting me know if it's not changing. Okay. So non-thermal, two types. So we'll be briefly seeing about photobiomodulation and photodynamic therapy. These are two therapies where non-thermal properties of light are used. And what is the source of light that is used? It can be either laser, which gives out collimated light, coherent monochromatic light with high energy and low energy. It can be used in both continuous and pulsated mode, and fine tuning of the tissue where you're working can be done. But there is a better, cheaper option. I wouldn't say better. There's a cheaper option of light emitting diodes, LEDs, which is becoming more and more popular now in phototherapy. So we'll first look at photobiomodulation.
Sorry to interrupt again. The slide is not changing, ma'am.
Okay, so is this fine?
Yes, it is fine now.
Yeah, all right. Thank you. I'll need my glasses now. Okay. So photobiomodulation. So when the effect of light is exerted on the living tissue, certain biochemical reactions happen, which can bring about changes in the tissue where you're treating. So here, deeply penetrating near infrared or infrared wavelengths from 600 nanometer to say 910 to 950 nanometer is used for treating local inflammation, pain. It is also known to promote healing, wound healing, and in some cases even periodontal bone regeneration. How this happens is there is increased upon the activation of the tissues using photobiomodulation, which is low level laser therapy. There is increased cell proliferation, there is improved blood flow, microvasculature blood flow, and there is increased cell proliferation due to the increased synthesis of collagen and procollagen. There's also angiogenesis and other modulation of macrophages and lymphocyte function happening. So these are some of the things that happen when photobiomodulation happens with low level laser. This all happens basically because there is improvement in the mitochondrial energy production. And basically, when infrared or near infrared light is acted upon mitochondrial enzyme cytochrome oxidase, there is increased availability of ATP and nitric oxide, which helps in the promotion of and production of increased growth factors and differentiation of mesenchymal stem cells, osteoblasts, etc. So I'll just quote few studies. This is an old study. This is probably one of the earliest studies in 2008, where a study was done to see if low-level laser, that is photobiomodulation, could it be used for new bone formation. It was done in an in vivo model, and they found that new bone formation was observed, cortical repair happened, healing of bone defect was seen, and they also found that when low-level laser therapy was applied on the surgical bed intraoperatively prior to the placement of bone graft, better results were found. This is one recent publication where photobiomodulation was done using 635 nanometer and 808 nanometer on human primary gingival fibroblast, and they saw that there was improvement in the viability, oxidative stress, inflammation markers, etc. by this study. They found that there was also downregulation of apotic related genes, and it modulated extracellular oxidative stress and inflammation markers. This is very recent, this year's systematic review, where they evaluated the effectiveness of low-level laser therapy, photobiomodulation on human bone healing, and it was found that bone healing was improved with the use of photobiomodulation. How did it work? Certain mechanisms which they have elaborated in the systematic review includes anti-inflammatory mechanism, analgesic effect, improved healing, and also as a result of these changes, the quality of life was also improved. Out of the 25 studies that they evaluated, this was the result found. Now, this was photobiomodulation. There is another treatment modality called photodynamic therapy, where light of a wavelength is used in the presence of a chemical, which is a photosensitizing dye. This has been used in periodontal treatment mainly as a substitute for antimicrobial therapy. So PDT, mostly antimicrobial PDT, has been used in periodontal treatment so far. And it is very important to make sure that the wavelength of light that you use corresponds with the absorption spectrum of the photosensitizing dye that you use. If you're interested in reading more about the light source and the dosimetry techniques applied in photodynamic therapy, because these are very technique sensitive, this is a good article by Michelle, which was done in 2020. This is a good read for you. So now if you want to look what is the role of photodynamic therapy in periodontal regeneration, as I said, mostly it was used as an ideal antimicrobial agent for treating periodontal infections. Now, its role in regeneration, very little is known about the role of PDT in regeneration, mostly antimicrobial therapy. Most of the publications which you will see that uses photodynamic therapy has been done on non-surgical, as an adjunct to non-surgical periodontal therapy, whether it is on healthy patients or on smokers, diabetic patients, patients with severe periodontitis, which is earlier classified as aggressive periodontitis. And all these photodynamic therapy in single or multiple has given better results in non-surgical phase. Now, when you use photodynamic therapy, that is light of a particular wavelength with photosensitizing dye, local conditions are very critical to get optimal results, because the effectiveness of the therapy is affected by the presence of biofluid. So if there is bleeding or presence of inflammation, this could interfere with the activity of the photodynamic therapy. So what you have to see is, when you're applying, you should minimize the manipulation of sulcus, the gingival sulcus, when you're irradiating, because if you manipulate too much, that will be bleeding, and then the efficacy of the treatment will be affected. So transgingival irradiation without inserting the tip of the probe into the gingival sulcus has been proposed as one method. And it has been found that in the presence of periodontal inflammation, multiple sessions of photodynamic therapy has reduced inflammation. Now, coming to the effect of photodynamic therapy in wound healing and its osteogenetic potential, this is a recent article where they found that low concentration of methylene blue was able to bring about wound healing, and also it promoted osteogenic related functions in both healthy and inflamed human gingival fibroblast. It also increased the expression of wound healing related genes. So there is some evidence of photodynamic therapies application in regeneration. Another article is photodynamic therapy, again with low concentration, that is optimal concentration of methylene blue, was found to improve the osteogenic differentiation, and it also had stimulatory effect on bone marrow derived mesenchymal stem cells. Although another photosensitizer was not effective, methylene blue was found to be effective on human mesenchymal stem cells in a low concentration. How these effects have been found is, the mechanisms of action explained in these literatures include inhibition of inflammatory mediators, the inactivation of host-derived cytokines. Especially this type of treatment would be useful for severe periodontitis, especially in medically compromised patients who cannot often go in for a conventional surgical therapy. And in cases where surgical therapy was done, especially in vivo animal models, they have found reduction in improvement in the bone healing, and also increased collagen production by fibroblast. Now, if if you were to combine nanomaterials with this light-based therapy, with phototherapy, you would see that most of it, or almost all of the studies, have been done in cancer-related therapies. So the issue, so if you want to apply light therapies in periodontal regeneration, there are certain issues that exist. First of all, there is a need for a well-established therapeutic phototherapy protocol, because these parameters that you use during light therapy, whether it's a concentration of the photosensitizing dye, or the wavelength, or the diameter of the probe that you're using, the optic fiber. All these are very critical. So a well-established protocol is very necessary, and there are limited number of clinically accepted photosensitizers that can be used, and there is a need for high target sensitivity, specificity, and again the available photosensitizing dyes sometimes lack proper tissue penetration, and the toxicity level also has to be optimal when you're using these for regenerative purposes. Another main issue that still exists, which could be solved in future, hopefully, that issue remains the unknown mechanism of the life-based tissue interaction due to its complex immune reactions. So in future, or the ongoing research focuses more on combination or incorporation of photosensitizing dyes into nanostructures. Already available studies show that such studies have been used, are going on in terms of methylene blue loaded polymeric nanoparticles, indocyanin green loaded nanospheres, carbon nanotubes, carbon quantum dots, graphene conjugated methylene blue. Then, of course, another interesting field that is coming up is gene engineering mediated PDT, and where there is genetically encoded photosensitizers. Another interesting field that they're working on is oxygen delivering nanomaterials and oxygen based nanocarriers, so that the efficacy of the light treatment that you're giving is improved. So when you see how low-level light can be used in periodontal treatment, there's a lot of ongoing research. There is no conclusive evidence, but a lot of ongoing research is going on, which shows that low-level laser therapy, phototherapy, is a promising tool in periodontal treatment, mainly owing to its anti-inflammatory and wound healing properties. There are few studies which show that applying laser even before the placement of bone graft could improve the healing property and the outcomes of the treatment. What you have to remember when you are using phototherapy is that parameters like energy density, duration, frequency of the radiation session, the wavelength that you're using, all these are very crucial parameters which need to be effectively combined for you to get the optimum result, because it is very important to make sure that these parameters are combined well to get the best laser tissue interaction. So these are my references, both old and new references. If anyone's interested, can take a screenshot. And there are a lot of new studies going on in this field. It's still it still is in the infancy, but there's lots to be done in terms of phototherapy in periodontal regeneration. Thank you so much.
Thank you, Dr. Betsy.
Thank you, sir.
That was a very sweet and small presentation.
Thank you.
I think there's one question in the chat box if you want to take it up.
Yes, please.
Nirmala is asking how long you need to irritate in order to reduce the inflammation.
How long you need to apply?
Yeah.
Yeah.
To reduce the inflammation.
To reduce the inflammation, it depends on the level of inflammation. You could give it in multiple sessions of 1 to 3 minutes, multiple sessions, along with removal of other causative factors would be effective.
Is this a reversible process?
Pardon?
Is it temporary reduction? Is it a temporary reduction in inflammation, or is a permanent reduction?
Because in the oral cavity, there is always occurrence of causative organisms which causes continuous inflammation. As and when the microbiologic load increases, there is again increase in the inflammation. So this has to be done on and off as the case requires.
Is there any other questions from participants? Okay, thank you, Dr. Betsy, for your overview on photomodulation, photodynamic therapy,
Thank you,
and the
sir.
usage of nanoparticles. I think the laser has opened up a lot of venues to use this. And I think nanoparticles, as you said, is usually in the cancer. Hopefully, it will be translated into the other fields also.
Yes, sir.
Uh, then thank you so much. I think I'll move on to the next session.
Thank you so much, sir.
Thank you. So, next session we have Dr. Anupal Lucas. She is an Associate Professor, Department of Community Medicine, Government Medical College, Kottayam, who will be speaking on the application of basic research in clinical scenarios, including various innovation in basic science, its application, role of in vivo models that enhance diagnosis, treatment, prevention, and control settings. So, over to Dr. Anupal Lucas for the next 30 minutes.
Good afternoon, everybody. Am I audible?
Yes, madam, please.
I'll try and present my PowerPoint. I think I have a problem sharing my screen. Can uh, can the organizers share my presentation for me?
Ma'am, just hold on, ma'am. We'll share the screen from here.
Okay.
Sure.
Yes. Can I start?
Yes, please.
Yes. That's my first slide. Thank you. So can I start? Am I audible?
Yes, ma'am. You're audible, ma'am. You may continue.
Okay. Just let me know when I am ready to go.
Ma'am, you can start, ma'am.
Okay. So, thank you to the organizers for inviting me for this talk. After listening to the first two presentations, I should say that I feel very out of place. I'm going to talk to uh, dentists, which is not my area, but I'll try. I hope the examples I give are not inappropriate. Okay, I have tried to incorporate something related to the dental area in this. But today I'm going to talk about research basically. So, research in dentistry or research in medicine, it's all the same. The basic principles are all the same. So, since I'm going to talk about translational research, I will first present you with this slide. What is translation to the and the original? So here is a picture of a lady who is in front of a lot of books, and I've written she is hardly working. And if I ask somebody to translate it for me, what is in your mother tongue? What does it mean? She is hardly working. What it means? Anybody? What does it mean? Anybody? Okay, I think the participants are all reluctant to answer. So I'll say she is hardly working to somebody who's not so for somebody whose mother tongue is not English, they might translate it into വളരെ കഷ്ടപ്പെട്ട് കഠിനാധ്വാനം ചെയ്യുന്ന ഒരാള് എന്നൊരു ട്രാൻസ്ലേഷൻ നമുക്ക് കിട്ടിയേക്കാം. സോ ബട്ട് ഇൻ ഇംഗ്ലീഷ് ഇറ്റ് മീൻസ് ആ ആള് കഠിനാധ്വാനം ചെയ്യുന്നില്ല എന്നുള്ളതാണ്. സോ ഫ്രം വെൻ യു ട്രാൻസ്ലേറ്റ് an original into another language, in very simple words, many things can go wrong. So, translation itself from original to real practical settings, translation of evidence into real practical settings is a very difficult thing. But that's not what I'm going to talk to you today. Next slide. I am trying to tell you whether the original is correct or not. Like I'm trying to ask you whether is she really hardworking. Okay, so it is possible that she's a really last minute person and this is the day before her exams and she's like, collecting all the books possible and going through the important questions and struggling. So it will look like that she's a very hardworking person. Actually, she's not. She's not at all hardworking. Or it could be that she's a very, very intelligent person and it's a day before the exam and if she has the capacity to go through multiple books and this is the first time she's seeing all these books and she will figure out what it is and she will get good marks the next day. So that brings me to the next question. Like, looks can be very deceiving. Similarly, when you're doing research, things can be very deceiving. So, am I measuring what I'm supposed to measure? Is she a really hardworking person? Am I measuring her hard work correctly? Or am I biased? Okay, have I studied her enough to say that he is hardworking, or it is just a snapshot? So, that is what brings me to the topic per se. So, the validity of what we are doing. So, what are we measuring? Did we measure it right? That is validity. Validity is measuring whatever we are trying to measure, are we measuring it correctly? That is called validity. Validity, next slide. Validity has two components. How does it apply to translational research? I will try and explain during this diagram. As clinicians, we are doing probably phase 2 trial or phase 3 trials where you do randomized control trial like Dr. Betsy was talking about phototherapy. So if I understand if she wants to generate some evidence in Indian setting regarding this, she will select patients, randomize them into two groups. She'll have one group whom she gives phototherapy. Am I right? The phototherapy is the right word. Is it the right word? Yes.
Yeah.
So you choose people with an infection, you give phototherapy, and other group you don't give phototherapy. So what she's doing is a randomized clinical trial somewhere in the middle of this picture. Okay. So so she has developed a protocol from her basic clinical research where some scientist or her co-workers has done some research in the lab and developed a protocol to do phototherapy. So they have done tissue research, they have done lab research and found out that if you use these dyes for this much duration of time, this will work. So now she has a protocol. She very much wants a protocol. So now she has a protocol, and she has a clinical setting, her medical college, and she has got patients with the disease. So she'll randomize them into two groups. She'll give one phototherapy to one group and phototherapy to other group and see whether it works. So where does this validity come from? And she has reached the conclusion that it works. So taking that evidence that it works, phototherapy works, and applying it to the population, the aged population or general population where there is an infection, and healing them and making their lives better. That is one part of translation. Translating what is the evidence which is generated in her clinic to the population so that the prevalence of periodontitis in the population comes down because of her phototherapy she gives in the clinics. That is one side of the translation. The other translation is all those people, scientists, basic scientists who worked in the lab and told her or generated a protocol for her is the other kinds of translation. So there is a translation from the lab to the clinical setting and from the clinical setting to the population. So I am going to talk about, since I am a public health person, I am not an expert in talking about the other side, but I can definitely talk about when you do an evidence before you generalize it and use it in a population. What are the things that you should make sure that what you have measured or what you have found out is really correct? You have measured it right. That is validity. Next slide. So this is something I picked up from the internet. I don't know whether it makes sense to you. So there is something called guided tissue generation, and I found that studies conducted by Neyman et al. Somewhere in the 80s, late 70s and early 80s, showed that only PDL cells have potential for regeneration of the attachment apparatus to the tooth. It doesn't make much sense to me. I don't know what PDL cells is. Uh, PDL cells, can someone tell me what PDL cells is?
Periodontal ligament cells, ma'am.
Okay, fine. So PDL cells have potential to generation of the attachment. This is a finding that the basic researchers have found out. Now somebody else has used it, and next slide, and done a study. Treatment of intra-bony defects with guided tissue regeneration in aggressive periodontitis, clinical outcomes at 6 and 12 months. So somebody has developed a protocol to use TCR and then treating this condition. So for that, they have used an RCT. So next slide. So when you have done a clinical trial like this, a randomized controlled trial like this, there is a truth in the study, whether you have measured it right in whatever study you have done, whether you have really found out the truth, whether you have gone wrong in some way, whether you have really found out the truth, and whether this is true when you're talking about the population. So I'll give an example like this. I want to measure the height of, I want to get the mean height of the medical students in my college, mean height of medical students. Okay. So I go and select 10 people. I go and select 10 people and I measure their height because I don't have the time to study 200 students. I go and study 10 people and I say, based on my 10 measurements, 10 people's height, I am predicting what is the mean height of the 200 people. Okay, so whether if measuring 10 students is enough to comment about the mean height of 200 patients is the truth in real life. That is one generalization. What is the truth in the study? I measured the 10 students, the height of the 10 students. Some students I measured them wearing a shoe. Some people I measured without wearing a shoe. Some people I measured by making them stand straight. Some were, leaning down or, not in the correct position. For some people, I use the correct instrument, stadiometer to measure. For our other half, some 5 or 6 people, my stadiometer was not faulty. So the mean height I got from these 10 measurements is totally wrong. Okay, that is the truth in the study. So truth in the study is whether when I measuring the height of 10 students, whether I measured it correctly. If I have measured half the people with footwear and half the people without footwear, my mean height is totally wrong. So there is no truth in what I have studied. And based on that false mean height, I am predicting the height of or the mean height of 200 people. So these are the two types of validity. Whether I am measuring the 10 people's height right is what's called the internal validity, and whether using this 10 people's height, whether I can predict the height or comment on the height of, not predict, comment on the height of the 200 people, that is external validity. So these are the two validities that we are talking about. So you must be appreciating the fact that, sorry, previous slide. You should be appreciating the fact that when there is any for a study, when you really want to work, if it really wants to work in the population, you need to have both internal validity and external validity. Another thing I want to say that if you don't have internal validity for your research, it is very unlikely that you will have external validity, because that there itself you have gone wrong. So whatever you do based on that is also going to be wrong. So no internal validity means that very likely, unlikely that you have external validity also. So now I'm going to talk about what all things you can do to have internal validity. To have internal validity, basically whatever you are doing, you should do it right. Like if Dr. Betsy does a study, like she said, she should have a proper protocol how to phototherapy is done. That protocol I'm talking about. Then she should randomize properly. She should select patients properly. She should measure the things properly. Outcome measures should be conducted properly. Outcome measures could be like, I don't know how she's going to measure the outcome, but outcome measures, everybody should be measured similarly. So there are lots of things that go into the internal validity of the study. So that is not what I'm going to talk about next. So next slide. Whenever we talk about, can you just put the next slide?
So next slide, ma'am.
Okay. There is another slide which is, can you just move it? There is a slide with internal and external validity. Sorry for this. Next. Okay, I think I've shared the wrong PowerPoint. Anyway, can I take 2 minutes and send you the?
Yeah, sure, sure.
It's okay, it's okay. I'll manage with this. I'll manage with this, whatever I have. So I'm going to talk about internal validity now. Okay. When you're talking about internal validity, that means doing your randomized clinical trial correctly. Okay. So let me start with each part of a randomized control trial. First is called randomization. What is randomization for? It is to address problem of confounding. So what is confounding? This is a simple example for confounding. I am trying to find the association between smoking and lung cancer. Okay. So there is a third factor, age, which is related to smoking. That is, I assume that when people age, they smoke more. So I have a third factor which is associated with smoking. How is it associated? Because as age advances, people smoke more. And age is another factor which is an independent factor for lung cancer. As you age, you have a chance of getting lung cancer, more chance of getting lung cancer. So age is a confounder which will distort the relationship between smoking and lung cancer. If I don't consider age in my study, I can go wrong when I'm trying to find the association with smoking and lung cancer. I will try and explain it with by phototherapy only. Like Dr. Betsy is she is doing a randomized control trial. For one group, she's giving phototherapy. For the other group, she's not giving phototherapy. Okay, imagine that she's doing that, and for some reason, she gives phototherapy to people who are younger and without diabetes mellitus for some reason. Maybe those young people consented to her study, or she is reluctant to give it to people with diabetes mellitus. For something made her give phototherapy to people with lesser age and people without diabetes. And she is measuring the outcome based on healing. Okay. So when she presents the study to somebody else, that person asks her, is it because of your phototherapy that they had better healing, or is it because you got a population who are much younger and who have got a population who do not have diabetes? Is that the reason why there was better healing? She doesn't have an answer. Dr. Betsy is still here. I'm sorry that I'm using your name in vain. I hope it is okay. Hello, Dr. Betsy? She's not there, huh? She left. Anyway, so here there is a study. The researcher is selecting a population who is generally younger in age and diabetes. For her own clinical reason, she's doing that. But finally, when she's comparing healing, if that group is getting a better healing, naturally the question will be raised to her: is it because of a phototherapy, or is it because of them being younger and not having diabetes? So the age and the presence of diabetes are confounding factors here. So how do we address this? There are many ways of addressing it, but in a clinical trial, the best way is to randomize. Randomization is a procedure in which I'm not going to go into details of that. Randomization is a procedure in which you do a you do a process. You do a process and you get two groups who are similar in everything, similar in age, similar in diabetic status, similar in compliance, similar in ethnicity, similar in race. Both groups are similar in everything, and the only thing that is different between the two groups is that one group gets phototherapy and the other group doesn't. So then you can say with 100% confidence that it is my phototherapy that caused the wound healing and nothing else, because everything else is the same. How you get the two groups which are equal in everything is called the process of randomization. And for those people who know about it, it is basically like a fair coin toss. It's a coin toss. I'm not going into details into that. So first of all, in an RCT, the heart of RCT is what I just told you, randomization. You randomize and you get two groups which are similar in everything. And you give an intervention to one group and no intervention to the other group. And it works. And you can be sure that it is that intervention that led to the healing and nothing else. Next slide. So this is what I told you. There is a study population. You decide on the inclusion criteria and exclusion criteria, and you have an experimental group and a control group where you give the intervention. And here the experimental group and control group and similar in everything. The only thing that is different between these two is that one gets phototherapy and the other doesn't. Then you get evaluated and measure the healing. That is the outcome. Next slide. So if I want to do a study to see whether paracetamol is better than a placebo in acute backache, I give paracetamol to one group and placebo to the other group. Both groups are the same in everything, same age. Both groups have equal number of females. Both group have equal number of diabetes. Both group have equal number of teachers. Both group have equal number of workers. Whatever. Only thing different is paracetamol. Next slide. Just go through the slides. Next. Next, next. Okay, this is the picture. This is the process of randomization. You have a bunch of people and you do the fair coin toss and you make two groups which are similar in everything. Like you can see that there are two girls there and two girls here. Two boys. Exactly, I'm not saying, but then randomization generates two groups which are similar. Next. No, how these are the slides which will tell you how to do this randomization. There are many techniques. Now we resort to computer generated techniques. The computer will tell you which is the patient comes to you and the computer will tell you whether this is the phototherapy group or the no phototherapy group. You don't have to do anything, but there are many ways in which you can do that randomization. Next slide. So these are some of the computer softwares which help you randomize and get two groups. Next. Okay. Now I come to the next part of the RCT, which is called allocation concealment. Like I want to use phototherapy. Okay. And I have a list of people who list of people who should get phototherapy and my control who should not get phototherapy. I am sitting in my clinic and I am allocating people to the two groups. I am the one who's decide whether this patient gets phototherapy and this patient does not get phototherapy. So I am sitting in my clinic. So a patient comes to me. He is an alcoholic. I know that he's not a compliant patient. He's an old age patient. He doesn't follow instructions. All that I know. That patient comes, and if I want my phototherapy group to do better, I am not very interested in enrolling him into in the phototherapy group. So I can do two things. Either I can not include him in the study, make him go away, or I can give the other control arm. I can put him in the control arm. Okay, so I am doing some manipulation here. So that will definitely affect the results. Whatever, once you have decided on the randomization sequence, the first person who comes to you, the first person who enrolls, should be given the treatment which is allotted to him by randomization. That means if it's decided already that the first person is getting phototherapy, then there should not be any manipulation. There should not be any possibility of him being excluded study, or him being given the no put in the no phototherapy group, so that I get the result that I am interested in or I am favoring. So what is the solution for that? The solution is that I should be concealed from the fact which is the next allocation. When the patient, alcoholic, non-compliant, aged patient walks into my clinic, till I enroll him and I give him the therapy, I should not be aware that whether he is going into the phototherapy group or no phototherapy group. So what can be done for that? Suppose I have 100 patients enrolled in my project. I have serially numbered 100 envelopes. And inside, serially numbered 1 to 100, I have numbered envelopes. And inside each envelope, I have written phototherapy or no phototherapy. So the patient comes in, I open the cover and I see phototherapy, I give phototherapy. Second patient walks in, I can't assess him or anything. If he's enrolled, I open the second cover, I read it. If it says phototherapy, I give phototherapy. Third envelope they open, no phototherapy, I give no phototherapy. So that process is called an allocation concealment. Allocation concealment means the person who is allocating the participant, the study participant into phototherapy group or no phototherapy group is not aware of the upcoming allocation. That is called allocation concealment. And how is it done? Next slide. I put it in envelopes. I take 100 envelopes, serially number them. I write the allocation in a piece of paper, put it inside the envelope, and keep it on my table. Next slide. I make sure that the envelopes are not transparent. So here is a teaching researcher who keeps the envelope across the tube light to see whether the next patient is going to get A or B. So if it is the patient, if he feels that it is a no phototherapy, if it's a phototherapy group, he puts in and once he realizes, he doesn't enroll that patient. So there, in spite of all these serially numbered, non-trans opaque envelopes, there is a possibility that the researcher might cheat and they allocate in the wrong way. So next slide. So I have already explained to you what is randomization, why is it done. Second, I have already explained to you what is allocation concealment, which are very two important things in clinical trials. Now I'll come to blinding. Blinding is a process. I am the investigator and I am assessing the effect of my phototherapy treatment by looking at the healing of the patient. Okay. So I go for my rounds or whatever. I see the patients in my clinic after the 7 days, and the patient comes to me, and I do an examination. And if I know that this is a patient who has received phototherapy, it is likely to affect my assessment, my clinical assessment, because I think that this works. I have a very positive feeling that it works. So when I do an examination, I am very likely to go wrong or go err towards the favoring phototherapy. So basically, if I am the person who is assessing the healing part, the assessing the outcome of the trial, I should not be aware who got phototherapy and who did not get phototherapy. So I am blinded. The patient, you take an informed consent, and if the patient knows that he is getting something new, so if the patient is a very apprehensive patient, if he feels that I am getting some new therapy, I don't know whether it works or not. So he is likely to the way he talks to you, the way he explains his symptoms, he explains his pain, it is going to affect him. So he should be blinded. The patient should not be knowing whether he is getting phototherapy or not. So I want to bring to your attention that there are certain conditions, certain situations where blinding the patient is not possible. If it is a drug, you can give them a cover, put the drug in a cover where there is no name or nothing. But for phototherapy, it is very difficult to blind the patient. So they, in surgical procedures, in some other procedures, it's sometimes very difficult to blind the patient and the investigator. But ideally, the investigator should be blinded as to who gets what. The patient should be blinded as to who gets what, and also the analyzing statistician should also be blinded as to who gets what. Okay, so that is about blinding. All these things should fall into place for an RCT to really work. Next slide. How much time do I have? I
Uh,
think I exceeded my.
yeah, yes, ma'am. You exceeded
Okay,
the time.
so that is about 30 minutes. It was.
Yes, ma'am.
What time is it now? 32. Okay.
Yes, ma'am.
So that is about RCT. Then RCT, I've only talked about, in a way, it is good that I send the wrong slides because I've only talked about internal validity now. I have a whole another set of 10 slides on external validity, but I don't think it's possible to cover that topic. So today I'll just summarize my presentation. I think you can stop the slide sharing. I was talking about what is translation. Doing translation is from the lab to the clinical setting. So what, and from the clinical setting to the population. So both ways, to translate it correctly, the original should be true. So how do you ensure that the original is true? That is whether you are measuring, where measured it right. So there are two types of validity. Internal validity is whether you have measured it right in that study group. So I say that phototherapy works, it should have really worked, that there is no bias. I've done my randomization correctly, I've done my allocation concealment correctly, I have done my blinding correctly, and that is the truth. Okay, that is internal validity. External validity is something whether the truth, when this principle, whether it always works. That will depend on my sample size. It will depend on my sampling. It will alpha error, beta error, confidence interval, all that. That's a whole other present another set of slides which unfortunately I can't tell you. But that is external validity. Okay, so that is my presentation in short. I hope I am able to convey something which is related to translational research in clinical, basic research in clinical scenarios. And once again, I thank the organizers for inviting me. Thank you.
Thank you, madam. I think what madam has done is reinforced the fact that we have to avoid bias in our research, and because a lot of bias come up in our research, and the fact that a lot of confounders are there, which we should recognize. So she did talk about the validity part. She talked about the randomization. She talked about the pitfalls in sample selection, and which we can overcome by concealment of the allocation and blinding. So, if there are any more questions on this, please do ask or write or type in the chat box so we can take it up. I don't see anything in the chat box, and I did not hear anything, so I think I will hand over to the organizers. Back to you. Thank you.
Thank
Thank
you.
you.
Thank you.
Thank you, ma'am. Thank you, sir, for the session and for the moderation. Now we move on with this. The lectures are all over, and now we are moving on to the poster presentations. Uh,
Good afternoon to all. We move on to the poster sessions. It's my privilege to welcome our judges for the poster session, Dr. Elna Paul and Dr. Gia Jose. Dr. Elna Paul is working as a scientist at ICMR Bioethics Department at Bangalore. She also serves as an adjunct assistant professor at Pushpuri Research Center. She is a PhD holder from Pukyong National University in Korea. Prior to this, she worked in the capacity of an assistant professor and researcher at King Saud University and Jazan University, Saudi Arabia. She has done her master's in oral pathology in London and graduated with her BDS from Bangalore. Her research interests include, but are not limited to, biomaterials, bone regeneration, stem cells, and osteointegration using implants. Elna ma'am, we welcome you to the session.
Thank you, Dr. Sharon. Thank you for your gracious introduction.
Dr. Gia Jose, she's a dynamic researcher with more than 12 years of experience in microbiology, biotechnology, nanotechnology, cell culture, and tissue engineering with around 38 publications and 3 edited books. Uh, Madam has She's a recipient of Senior Research Fellowship by Council of Scientific and Industrial Research (CSIR). Dr. S. Kothari Postdoctoral Fellowship by the UGC, Postdoctoral Research Fellowship from Stockholm University, Sweden, and Research Associate Fellowship from the ICMR. Currently, she is the Research Associate with ICMR, Cochin University of Science and Technology, Kochi, Kerala. Gia ma'am, welcome to the poster sessions.
Thank you, ma'am. Thank you for the nice introduction.
I welcome all the participants who have logged in. Shall I start? All the participants, kindly note that the posters and the audio clips that you have sent have been downloaded in our system, and we'll be presenting it or sharing the screen from here. In case of any technical glitches, kindly be ready to present from your side also. Madams, shall we move on to the presentation?
Yes,
Shall
of
we
course.
begin?
Yes, yes, please.
Fine. Can you see if it's there now? Is Mr.
Yes, it is visible. It is visible.
Respected dignitaries present here, myself G. Meenu, third year postgraduate student from Department of Periodontics from Pushpuri College of Dental Science, Tiruvalla. Today I am here to present my poster titled In Vitro and In Vivo Biocompatibility Assessment of Demineralized Fishbone Internet for Periodontal Regeneration. Periodontitis is a common inflammatory condition and it's a major cause of tooth loss in adults. New attachment periodontal regeneration is ideal outcome of periodontal therapy and bone grafts are widely used in bone regenerative procedures. And xenografts are a section of tissue from one species grafted onto the different species and bovine bone is a commonly used xenograft for bone regenerative procedures. And a major concern with these bovine derived product is the potential transmission of zoonotic diseases and also prion infections such as bovine spongiform encephalitis. Hence, marine organisms are received increased attention in bone regeneration due to its extra advantage of lack of disease transmission in humans. Hence, the objectives of the study is mainly to fabricate and to assess the in vitro biocompatibility, which is in terms of cytotoxin and also cell proliferation, and also to assess the in vivo biocompatibility of demineralized fishbone in NNet for periodontal regeneration. Coming to the methodology, it's mainly we're assessing the cytotoxicity of demineralized fishbone using MTT assay. Here the fishbone, that is 10 milligram per ml, was added to the cells and MTT is further added and the presence of violet formazole can be analyzed using an ELISA plate reader. Then coming to cell proliferation using scratch assay, a scratch was made in the middle of every well with a pipette tip and extracts was added in each test well and a control well. Then we'll proceed with the in vivo biocompatibility assessment. The demineralized fishbone is implanted subcutaneously in male scratch controllee rats. Coming to the results showed that the demineralized fishbone displayed a favorable tissue compatibility without any abnormal response. And the in vitro assessments also proved that they are not cytotoxic with excellent cell proliferation. And the in vivo biocompatibility testing in animals showed that they elicited no unfavorable tissue reaction and is biocompatible. And the study finally concluded that the demineralized fishbone showed a superior biocompatibility as compared to the commercially available oceanograph, and hence it can be an effective biomaterial for periodontal regeneration. Thank you all. That was the first poster. We move on to the second poster. Before that, is Dr. G. Meenu on online? Is she on the meet? Would you unmute yourself, please, for the question and answer round session? Yes, Dr. Meenu. Judges, if you have any questions for the presenter.
Hi, Dr. Meenu. That was a lovely presentation, I must say. If I may ask, which fish bone or which fish did you choose? And was there anything, unique about this particular fish that you chose?
Actually, I chose tuna fish, the backbone of tuna fish. Uh, am I audible,
Anything?
ma'am?
Yes, yes, yes. Anything special about tuna fish that you think would help in?
Tuna, well, we have like this less cytotoxicity and also it was
Okay, thank you so much.
Dr.
Thank you.
Nino,
Okay, ma'am.
am I audible?
Yes, ma'am.
Yes, that was a nice presentation. And my question is like, which method protocol you used for the demineralization? There are several protocols. So which one you selected and
Yes,
why?
ma'am. Lyophilization, ma'am. Lyophilization. Ma'am,
Sorry,
lyophilization method.
lyophilization.
Method.
Sorry, actually I didn't hear you.
Lyophilization, ma'am. Lyophilization.
No. Actually, I'm asking about the demineralization protocols.
Ma'am, actually, the demineralization was done by the so at Central Institute of Fisheries and Technology. We got a demineralized bone, so demineralized fish bone from there. Then we proceeded with the following experiments, like in vitro and in biocompatibility assessment was done.
Okay. Okay, thank you.
Thank you, ma'am.
Thank you, Dr. Minu. We shall move
Thank
on
you,
to
ma'am.
the next poster.
Five. Respected dignitaries present here, myself Jimmy.
Sorry.
Here to present my poster on the topic evolution of regenerative therapy in the industry. Periodontal disease is the most common disease of oral cavity which leads to
Sharon, I'm afraid we can't see the poster.
industry is constantly evolving and
Ma'am, you're not able to see the poster, ma'am?
Yes.
Just a second.
Yes. It's fine now.
hello everyone, today I'm here to present my poster on the topic evolution of regenerative therapy in the industry. Periodontal disease is the most common disease of oral cavity which leads to attachment loss and bone loss. Regenerative dentistry is constantly evolving and expanding. Younger attack in 1993 proposed tissue engineering in periodontal tissue as a possible technique for regeneration. The tissue engineering contains three key factors which are dental stem cells, scaffold, and signals. Dental cells which have the ability to differentiate. Example, periodontal ligament derived cells, periodontal ligament mesenchymal stem cells. Scaffold here provide physical support for the healing area and also serves as a three-dimensional substrate for cellular adhesion, migration, proliferation, and production of extracellular membrane. Example are endosan and polyglycolic acid. Now moving on to signaling molecules plays a role in modulation of cell-to-cell signals and cellular activities in development and maturation of the cell. Example are protein-derived growth factors, more morphogenetic proteins, mRNA in gene therapy, etc. Some of the outcomes from tissue engineering are injectable delivery system, the nanocapsules or membrane, periodontal tissue reconstruction by in vitro culturing, then recombinant patient-derived growth factors, which is also known as GEM21. Let's discuss some common biomaterials used in periodontal regeneration. Bone grafts. Bone grafts are used in bone regeneration and gold standard for that is autografts. Gingival tissue regeneration is used because of the turnover rate of the epithelium is more compared to the turnover rate of periodontal tissue. To prevent the invasion of the epithelium to the periodontal area, the gingival tissue regeneration was introduced. Moving to enamel matrix protein, for example, for there is endogame, it contains amylogenins which involve in development of enamel and periodontal attachment. Platelet-rich plasma. Initially, it was introduced as PRP and use of antiviral, which it caused delay in wound healing. Then it was modified into PRF. Amendment in field of regeneration therapy is 3D bioprinting. It contains of three type and it is used in development of custom implant, dental pulp complexes, fixed dentures, craniofacial skeletons, etc. The three types are inkjet bioprinting, micro extension bioprinting, and laser bioprinting. Thank you.
Dr. Anjali, if you are online, would you kindly unmute yourself? Dr. Anjali?
Yes, ma'am.
Judges, the session is now open for questions.
Thank you, Dr. Anjali, for a wonderful presentation. Out of the many
Yes, ma'am.
different out of the many different formats or the different methods or the different advances, can you hear me, Dr. Anjali? Hello, Dr. Anjali, can you hear me?
Yes, ma'am.
Yeah. Of the many different
Yes, ma'am. I can hear you.
of the many different advances that you've explained here that can be used as a regenerative therapy, would you like to tell us more about dental stem cells?